I'll be introducing you. And then we've got Mark McPeak as our broadcaster for ensuring that everything goes smoothly with Zoom. And then Nitin is going to be our questioner. So I just want to note that our 4:40 has canceled due to illness. And so we'll just take a break at 4:40 if you're planning to stay the whole time. This is the second day, so you probably know, but each speaker has 20 minutes, intended to be about 15 minutes for presentation and then 5 for questions. And I will let you know when you have 5 minutes left by raising my hand virtually. And then when you have 3 minutes left, I'll make a note. And at 20 minutes, I'm going to end and introduce the next person. As a reminder, please keep your mics on mute during the presentations. If you have questions, you can ask them via the chat or the Slack channel. And then once the presenters are done, then Nitin is going to broadcast the questions to the speaker. So without further ado, let me introduce the first person. And while I'm introducing you, feel free to start sharing your screen. So up first, we've got Jimmy Peniston, who's going to talk about the role of ecological interactions during adaptation to environmental change.
Awesome. Thanks, everyone. My name's Jimmy. My pronouns are he, him, his. And I'm excited to talk about the role of ecological interactions during adaptation to environmental change. If I get my slides to work. Before going any further, I also want to acknowledge the fact that this is all part of my dissertation work at the University of Florida, which is located on the land of many nations, including the Seminole and Timucua people. And I want to also position myself as someone of European descent working within academia to study ecology and evolution. And I think this is particularly important because there are alternative ways of studying nature that I wasn't taught about were valid, but they are equally as valid. And this is particularly relevant for someone like me who uses mathematical models to study nature, I think, because we think our models are completely objective, but in reality, my work is really influenced by colonialism and society at large, and the assumptions I make, the questions I ask, and the language I'm going to be using to talk about this. So it's something I just want to preface my work with because I'm continuously trying to unpack that. I just want to preface that before I go further. So with that being said, I'm really excited to talk about a topic that's really important to me, which is how evolutionary biology can inform conservation biology. In particular, I'm interested in rapid evolutionary responses to environmental change and anthropogenic change. So where these changes are climate change, pollution, or the introduction of non-native species, we have really good examples of rapid phenotypic evolution that has demographic consequences for the populations being affected. And I don't have time to go through all these examples, but there's lots of really good examples. These are all from vertebrates, but also from inverts and plants that show that evolution can really matter for how these populations will react to anthropogenic change. And in particular, I'm really interested in really dramatic environmental change that moves a population outside of its ecological persistence limits, so outside of its niche per se. And this falls in within the framework of evolutionary rescue theory. And to me, evolutionary rescue is simply the question of when can evolution save species from extinction? More formally, if you want it that way, it's when evolutionary rescue occurs when local adaptation rescues a population from demographic declines caused by environmental change that would otherwise lead to local extinction. So graphically, what's going on here is if we have, there's an environment before the environmental change and there's some optimal phenotype, which is here, and then the environment changes and that optimum phenotype changes, right? And I'm gonna be modeling an abrupt change in the environment, but people also model linear and continuous changes of other forms. Demographically, what happens in response to this is before the environmental change, the population is at carrying capacity or fluctuating around there. And then after the change, the population with no evolution will plummet to extinction because it's maladapted. With evolution, the population will decline to extinction, but if it adapts quick enough, it can evolve to have a positive growth rate and then actually persist. But I showed 2 lines here, one where the population adapts quickly and one where it doesn't adapt, where it adapts more slowly and is at a low population size, or it's at high risk of extinction. And I point out this line in particular because in many evolutionary rescue models, we can't actually, the way the modeling works, the population size can never actually go to 0, which is kind of not optimal if you're interested in extinction. So the way we model is we have this arbitrary cutoff, and we say below that population size, the population is at a high risk of extinction. So this red line was at a high risk of extinction. So there's been lots of theory on evolutionary rescue, trying to figure out which factors make it more likely for a population to adapt or extinct. And some things we know are really important are the initial population size, the degree of environmental change, the additive genetic variance, things like that. Last thing I want to say in general about evolutionary rescue is that this work has actually been really well supported empirically, the theory has been. So Graham Bell and Andy Gonzalez have done a series of experiments on yeast adapting to salt that supported the theory well, and Ramsayer and other people have looked at bacteria adapting to antibiotics. So what we wanted to do in this project is ask the question of how do ecological interactions affect evolutionary rescue? And by we, I mean my co-authors and I. So my co-authors are Mike Barfield, Bob Holt, and Richard Gomikowicz. And without them, literally couldn't have done most of the math I'm going to show. So they were part of all of this. And I also want to acknowledge that we are not the first people to ask this question of how do ecological interactions affect evolutionary rescue. So there's been work by Matthew Osmond and colleagues, and Graham Bell and his colleagues on this, and a few other papers on these lines. And there's also a whole emerging literature on community rescue, which has looked at how whole communities respond to a single environmental stressor. I'm going to focus today particularly on predators and facilitators and how they affect evolutionary rescue. So first, for predators, how do predators affect evolutionary rescue? I'm going to show this diagram where the focal species is going to be this butterfly, because I like butterflies, and there's some environment that's affecting it, right? So here I have, there's some environment affecting the focal population, and this would be the typical evolutionary rescue model. Is just looking at this. We have an environment that's going to change that affects the population. And the question is, what happens when you add a predator in? And some people have looked at this, and they've typically looked at when there's just selection on this environmental trait, right? So the predator is just having this negative demographic effect on the predator. And what you generally see is that the predators negatively affect evolutionary rescue, which makes sense. It makes it harder to evolve when you have more mortality. And we see that in our models too, which I'll show. But I also want to highlight that there are multiple papers, but in particular this one by Matthew Osmond and colleagues in AmNat in 2017, that show places where predators can actually facilitate adaptation, evolutionary rescue, which is when they either preferentially eat the more maladapted individuals, or they're releasing the prey from negative density dependence. We're not going to see those interesting things, those specific interesting results in our model, because we have a more simple demography. But what we wanted to add to this literature is ask, what is the effect of genetic correlations between ecological traits? And so to show this, we're going to have the same diagram, but instead of just having this trait, there's also going to be selection on the attack rate. And there could be a genetic correlation between these two traits. And Mellard et al. Looked at in 2015, they slightly got this effective genetic correlation, but they only looked at these extreme cases of either perfectly correlated or perfectly uncorrelated. So they didn't look at any in between on genetic correlations. They also used an adaptive dynamics framework, which assumes that the population is mutation limited. And we're going to use a quantitative genetic framework to look at this. Okay, so just to show what's going on in these models before I get to all the math is, before the environmental change, there's going to be, both traits are going to be well adapted. So the population, so both the predation and the environment are both, the population is going to be well adapted to them. But after the environmental change, the optimum phenotype for the environment's going to change, but the optimum phenotype for the predator, the attack rate, is going to stay the same, right? So it's going to be, the population is going to be well adapted for the predator, but it's going to have to adapt in order to persist, in order to be adapted for the environment. So mathematically, how we're doing this is we have discrete generations with no density dependence and a generalist predator. And this is just exponential growth. And this R term is it's an intrinsic growth parameter. A is the attack rate parameter, and P is the predator density. And one trait, the first trait, X, determines this growth rate, right? So this is the environmental trait. And what this is saying is the farther the population is from this optimum phenotype, theta, the lower the growth rate. So this is a Gaussian fitness function, and this gamma term determines the strength of selection. For the second trait, Y, which determines the attack rate, we have the same thing, but the more maladaptive the population is, the higher the attack rate. Okay, so these next few slides have a lot of math. Sorry, go through them quickly. But we can take these two equations, put them in here, and get the fitness, which is this right-hand term, which is W here. And we can rewrite it in this other form. And interestingly, what comes out is the strength of selection on the Y trait is dependent, so on the attack rate trait, is dependent on the predator density. And it's confusing because this is inversely related to strength of selection, but what this is saying is the higher the predator density, the higher the strength of selection on the trait related to attack rate, which makes sense, but this comes out of the model, which is reassuring. Anyways, then we can rewrite this all in matrix form, which I won't go through too much. And that's useful because then we can use this machinery developed by Gummicowitz and Wohl to find the mean fitness of the population is this. And the one things I want to highlight here is this is, what comes in here is the phenotypic variance covariance matrix, the G matrix, which you've probably heard of, the genetic variance covariance matrix, and the environmental variance covariance matrix. Okay, so with all that, sorry for going quickly through that, here's the full set of equations, which tells us the mean fitness of the population given the two traits. There's a vector of traits, how that affects population growth, and how that, and then also how the population phenotypes are going to change over time, which is determined by the genetic variance covariance matrix and the selection gradient. Okay, so the first thing I'm going to show here is just the result with no genetic correlation between the traits. And this is just showing how adding predators hampers evolutionary rescue, which is what other people have shown too, and makes sense, right? So what you're seeing here is with no predators, this is what the trajectory looks like. And when you add predators, the populations go to lower population sizes and stay there longer. So they're at a higher risk of extinction. So now what's the effect of genetic correlation? We have the same line. This is P equals 4 from over here, just showing it for longer. And when you add in more genetic correlation, that further hampers evolutionary rescue, right? And that's because the population is being held back from adapting to that environment by the correlation to the attack rate trait. So to summarize the effect of predators, both the demographic effects of predators hinder evolutionary rescue, and so do genetic correlations between these ecological traits. So the take-home message here is if we're ignoring predators altogether, we're probably over-predicting the probability of evolutionary rescue. And even if we're considering the demographic effects, we also might be ignoring the genetic correlations that might matter a lot as well. So I just want to take these last few minutes to talk about some work in progress on looking at how facilitators affect evolutionary rescue. So if instead of having something that negatively affects the predator or the focal population, like a predator, what if we have something that positively affects it, like a host plant or some sort of resource? So I'm not going to go through all the math for this because it's basically identical to the other math, besides there's a plus sign instead of a minus sign a few places. But what we see here is these are the demographic effects of adding facilitators. They're called, I have them M here. That's because I was incorrectly referring to them as mutualist, but they're facilitators. And so as you increase the facilitator density, it facilitates evolutionary rescue, which makes sense. This is with no genetic correlation. And when we add in genetic correlations, though, we still see the same negative effect that we saw before with the predators, because it's hampering adapting because there's different optimal phenotypes between the two. But what's interesting, concluding when you put these together, is you can look at these two different plots where one is with no genetic correlation, right? So as the plot I showed before, we're adding facilitators, facilitates rescue. But with a genetic correlation of 0.75, we get this case where with no facilitators, it's the same. But then if we add in some facilitators, it actually has this negative effect, right? Where the population goes to lower population sizes and it stays there a little longer sometimes. But then if we add in even more facilitators, it becomes beneficial again. So I think it's interesting that we get this effect that was this, it always was beneficial with no genetic correlation to add facilitators in, where with the genetic correlation, it's actually, it becomes this nonlinear, non-monotonic effect. So the last thing I want to say is we only looked at a couple scenarios here, right? Where we either had one predator species or one facilitator species. But in nature, a lot of populations will live in are more complex environments, right? Where they'll have both a predator and a facilitator, and they'll have multiple environmental traits that matter. And these will demographically affect the focal population in different ways. But there's also the chance that, well, there's also going to be selection on all of these characteristics, and there could be genetic correlations between the traits that matter in all these scenarios. And what I think our work is hinting at is that this could mean that if we're not considering genetic correlations, we might be overestimating a lot of time how well populations are going to respond to environmental change. But I also want to point out that there are times when genetic correlations could be helpful too if the optimum phenotypes are in different directions. So there's still more work to be done on that case, but I think this is suggesting that it's something we should be considering more in these models. So with that, I want to once again acknowledge my co-authors, the University of Florida and AMNAT, for putting on this conference, and I'm happy to take any questions. Thank you.
Thank you, Jimmy. That was a great talk. We have time for a few questions. So if you have any questions, please type it up either on Slack or Zoom here. Yeah, looks like there are no questions so far. Yeah, sorry, there are a few. Maddie Gamble asks if you modeled evolutionary rescue with negative genetic correlations.
So no, we did not, and that would be, I think, the intuition would be it would then be potentially beneficial, which would be an interesting. That's a very good point. I don't know. I just didn't look at that. Mathematically, it's completely possible in our models. Just have to change the parameter, I'm pretty sure, to be negative. So that's a good point, and we haven't looked at that, but I will after this. Thank you.
Yeah, the second question is from Gil Hendricks. Great talk. Did you track the phenotypic traits over evolutionary rescue? Did the populations abandon either the predator or facilitator optimum or the environmental optimum?
That's another good question that I can also do, but also did not do, which is I think it would depend on the strengths of selection on whether or not they would abandon one of the optimums or not, or find some in between ground. And that will depend on the parameter values, but that's an interesting thing, and I can definitely look at that. Thanks, Gil, for that question.
And next question from Sam. The model is deterministic. How might stochastic variation change this?
That's once again a good question. I don't think my intuition would be it won't matter much, the stochastic variation, although I could be wrong. As Sam probably knows too, I plan on building individual based simulations that are stochastic to pair with these, which I often do. And most of the time, the results are surprisingly similar. I think what would matter more than the stochastic variation are some of the other assumptions that are built into this model, like constant genetic variation and phenotypic variation, how that doesn't change over time. And I think that, to me, seems like a more inaccurate representation. But there'll probably be some effects of stochastic variation, but I don't see it being in any particular way. But I haven't done it.
Yeah, thank you, Jimmy. In the interest of time, I don't think we'll have any more time to take questions, but there are a few questions. If you can answer that by typing into Zoom, I think that'll be great. Thank you so much.
Yep, thank you. Thanks, everyone.
Yes, thank you, Jimmy. And next up, we've got Renee Petipas talking about Back to the Future, using herbarium specimens to study coevolution. You need to unmute yourself, Renee.
Can you hear me now? And you guys can see my screen okay? Okay, great. Let me just turn on my laser pointer here. Having technical difficulties, obviously. So I'm really happy to be here. Today I'm going to be talking about methods I'm developing so we can eventually use herbarium specimens to study coevolution and plant microbe interactions. This work was done in collaboration with Amanda Antoch, who helped me with various aspects of the project, but especially the culturing experiment I'm going to talk about. And this work was done in Maren Friesen's lab at Washington State University. So coevolution is reciprocal evolutionary change between interacting species. So on this slide, I've included some canonical examples, including yucca and yucca moths in the top left corner, acacia trees, which are in the background here, and presumably they're associated acacia ants in the top right corner. And then down here on the middle panel are some plant roots with nodules they produce, and within the nodules are their symbionts, which are nitrogen-fixing bacteria. Coevolution is a dynamic process that's important because it probably has shaped the diversity of life, including even beetle diversity, which is one of our most diverse taxonomic groups. So when evolutionary biologist J.B.S. Haldane was asked what we can conclude about the nature of the creator, he famously replied, "They have an inordinate fondness for beetles." And in this paper from the late 90s, the authors concluded that beetle diversity was perhaps driven by coevolution between beetles and angiosperms. However, studying coevolution is difficult because we need to fulfill this definition of reciprocal evolutionary change, which is especially complicated for macroorganisms. We do have methods that we can use, including phylogenetic methods as they used in the beetle paper, where we might look for congruence in phylogenies to detect evidence of coevolutionary diversification. We can also use space for time substitutions, which assumes that populations are at different points in their coevolutionary history, and thus can be used to understand coevolutionary trajectory. And finally, we can do experimental evolution, which has been done with great success with microorganisms. This figure comes from a recent paper out of the Seagraves lab, where they experimentally evolved a yeast mutualism to understand coevolutionary processes. But there are still special challenges with studying coevolution for macroorganisms. So today I'm going to propose that we can use herbaria, which are collections used to preserve and store dried plants, as a resource for understanding coevolutionary processes and plant microbe interactions. So in the Friesen lab, we work with a group of plants called legumes and their root-associated nodule-producing bacteria called rhizobia. Rhizobia fix nitrogen for their hosts, and in turn, their legume hosts provide carbon resources. We can use herbarium specimens to study this relationship because specimens often have seeds and roots attached. So here on these two herbarium sheets, we have Metacoga lupulina, which is the focal plant species I work with. You can see the seeds in black here and the root mass at the bottom of the sheet. And if you were to zoom in, you would be able to see that there are nodules attached to these roots. So on this panel on the right-hand side where the arrow is pointing, we see a nodule that we've rehydrated. This specimen, these roots are collected from a specimen from 1930. So you can see when you rehydrate it, the nodule plumps out and starts to look a little more like the nodules I showed in one of my first slides. So in this talk, I'm going to focus on methods I've developed that we can use to eventually study coevolution and plant microbe interactions. I've been using a combination of culture-dependent, culture-independent, and plant trapping approaches to recover and describe historic bacteria. I'm mostly going to talk about these culture-dependent approaches, but I'll briefly touch upon the idea of plant trapping. So during my postdoc, I've done 2 major herbarium samplings, each using specimens from Washington State that were collected in Washington State. The earliest specimen that I've worked with was collected in 1925, and the most recent was collected in 2015. This work falls under the category of resurrection biology because I've been trying to revive and characterize bacteria growing in specimen nodules. But eventually I'll also be doing some genomics work, which will allow me to answer a complementary set of questions. So today I'll focus on this first set of experiments where we use 7 solid media types to try and isolate bacteria from specimens collected at 5 time points. I'll also share some results of some Sanger sequencing. We've sequenced about, a little bit less than half of our collection at this point. And then we selected a subset of these cultures and we used them to inoculate plants to understand how they affect plant phenotype. And lastly, I'm going to talk about a pretty different approach that we took where we let the plants take over the heavy lifting of waking up the quiescent bacteria. So for these plate culturing experiments, we collected 1 nodule from specimens that were collected in 2015, 2004, 1986, 1974, and 1950. And we compared these with contemporary plants that were collected in Pullman, Washington. We selected 1 nodule per sheet. We rehydrated it. We rinsed it 7 times with autoclaved Millicue water. We crushed it, and we plated it on 7 different media types. We use 3 media types that are typically used in microbiology labs, including tryptone yeast, yeast mannitol, and Luria broth. We also made modifications to the yeast mannitol, which is frequently used to cultivate rhizobia. So we diluted it in order to control the production of free radicals. We added salt in order to help with osmotic balance. We added calcium, which can strengthen the cell wall of rhizobia, and we added pyruvate, again, to help with oxidative damage. We also collected a similarly sized leaf fragment, and the idea behind this is we wanted to control for environmental contamination that would be coming from the herbarium. As you can imagine, herbaria are dusty places full of a lot of plant material. So we wanted to make sure that the rinsing procedure we used on the nodule was removing exterior contaminants. So we put our leaf fragment through the same pipeline here and plated it out on the same media types. We've also collected seeds, and we've done a number of germination experiments, and we now have historic genotypes of the focal plant species Metacoga lupulina. I won't talk much about that work today, but I'm happy to answer any questions. So through our culturing work, we managed to get 109 strains to grow. And this was from specimens collected in 3 different years. We got strains growing from 1950, 2004, and 2015. We didn't find an interaction between the year the specimen was collected and the media type. So here I've just shown all the media types together, all the years averaged together. So on the y-axis, we have the probability of growth, and on the x-axis, we have the different media types. There's no statistically significant influence of media type on the probability of growth, although we do see this bump in TY, which I'm going to go back to later. So these results indicate that historic strains can be revived with media typically used in plant microbiology labs. So we've Sanger sequenced some of these strains. And as I indicated earlier, we were really interested in trying to get rhizobia from historic specimens in culture. So there's, in particular, Ensifer species that associate with Metacago lupulina. Unfortunately, our Sanger sequencing hasn't indicated that we have any of those in our culture collection, but we do have a pretty diverse range of bacterial taxa. We ended up with a lot of bacillus, which is probably not surprising given that they're spore-forming bacteria and they can probably withstand the conditions in the herbarium quite well. We also found good representation of an interesting group called Microvirga, which is in the order Rhizobiales. Microvirga has been demonstrated before to produce nodules, and there's some recent work by Ali Igwe that shows that they're common in serpentine systems. So we were interested to see this group in our culture collection. We were also interested because as part of our collection, we're creating a database where we're recording the morphology of the bacterial species as well as taking pictures. And we could notice immediately that some of the strains that we were getting in culture look quite similar to Ensifer medicae. So this panel on the far right is from one of our contemporary specimens collected from Pullman, Washington. So they were isolated from a contemporary nodule. And they look very similar to these Microvirga species that we're getting out of the historic nodules. So we decided to take some of these strains from 2004, some of these Microvirga strains from 2004 and 2015 to carry them into an inoculation experiment to see what effects they have on the phenotype of the plant. So the idea behind this is we really want to fulfill Koch's postulate. So anyone who studies microbiology is familiar with Koch's postulates. And the idea here is that we can establish causality between a microbe and a host phenotype of interest. So in our case, we have isolated microorganisms from herbarium specimens, grown them up in pure culture, and we're going to take these pure cultures and inoculate the same host species and see if we can produce the phenotype of interest. So in our case, that phenotype is nodules. So in order to do these inoculation experiments, we used contemporary genotypes of Metacago lupulina collected in Pullman. We created an inoculum using Microvirga strains from 2004, 2015, and we compared these with strains isolated from contemporary nodules. We inoculated plants in the sterile growth system that we developed, which involves growing plants in vermiculite in glass tubes and using micropore tape. And here we really wanted to ask 2 questions. We wanted to see if any of these strains nodulate, and if they nodulate, if they benefit the plant. So we found that the historic Microvirga strains from 2004 do nodulate plants. The 2015 strains, we didn't see any nodulation. We found when plants have nodules that they are bigger than the non-nodulated plants, and that the 2004 strains of Microvirga benefit plants equivalently to the 2019 fresh strains. And we also compared this with a lab strain of Ensifer medicae called WSM 419. Now I'm going to talk quickly about the plant trapping approach that we took. So the idea here is we wanted to take advantage of species-specific signaling that occurs between Metacago lupulina and rhizobia. We also thought that maybe the rhizosphere of the plant is a more hospitable growth environment than the harsh interface of the microbiology plates for the rhizobia. So here we just used a similar method as we used in the previous experiment. So we collected 1 nodule per sheet, and we used a wider range of years here. So the most recent specimen we looked at was 2015, and the oldest specimen we looked at was from 1925. And again, we compared these with fresh, with contemporary isolates. So we took 1 nodule, and in this case, we followed similar protocol where we rehydrated it, we rinsed it. In this case, we actually used a light bleach solution. And we added an additional step where we incubated the entire nodule overnight in a shaker incubator. We then rinsed it and crushed it, and we allowed the crushed nodule to grow up overnight. And then we directly inoculated plants with this liquid material. And in this case, we only used dilute TY because we wanted to take advantage of that bump that we saw in the first culturing experiment, where it seemed like things were going a little bit better with TY media. So because I am now not just an academic, but I'm a pandemic academic, the results of this, we just finished this experiment on Friday. So I have not analyzed the growth results yet, or the mortality results, to see how these different cultures may affect those aspects of plant phenotype. But we did look at the root systems of the plants to look for nodulation. So initially we were really excited. These 2 pictures on the left are from a historic specimen from the historic nodule. We were really excited, thinking that this was nodulation. So we took some pictures using our dissecting scope. And when we look at the historic, I'm not really sure what to call them, bumps on the root, they don't look quite like a nodule. They are some sort of outgrowth, and I would be really interested to, maybe it's a nodule that started to develop but didn't finish. And you can compare this picture on the left with, this is one of our control plants, and this is sort of more what you would expect with nodulation. So this is definitely still a work in progress, and we have a lot of follow-up work to do, but sort of an interesting finding here. So just to conclude, we can culture bacteria from herbarium specimens using growth media commonly found in labs. We don't have any indication that we're recovering any Ensifer species, which is unfortunate, but maybe we'll have more luck with different approaches. Some historic bacteria that we're recovering do nodulate plants and are beneficial. And trapping might be a way to recover additional bacteria, but we really need to dig more into these results. So this is still research in progress. We have to finish sequencing our culture collection to see what else we have. Because we have contemporary and historic seeds of plants, as well as contemporary and historic microbes, we'd like to do a time shift experiment where we're comparing the compatibility between these historic and contemporary bacterial strains and their hosts. And finally, I'm hoping the system can eventually be used to test some of the principles of the geographic mosaic of coevolution. And that's all for today. So I just want to thank members of my lab, including Maren Friesen, for being an amazing advisor. This work was really done in collaboration with Amanda Antoch, and Drew Rothy helped a lot. I'd like to thank the Marion Ownby Herbarium for giving me access to the specimens. And this work was funded by the National Science Foundation. I was a collections fellow. And I want to thank the American Society of Naturalists for giving me an opportunity to speak. Thanks, and I'll take any questions.
Thank you, Renee, for the wonderful talk. We have time for one question. If you have any questions, please post it here. Yeah, Maria asks, do you remove eukaryotes in some way?
We haven't done anything like that. I guess you mean like fungal contamination on. Is she there?
And no, her follow-up question is if you're getting any nematodes in your samples or not.
Oh yeah, that's it. We don't do anything to control for that. We use very sterile methods and a lot of negative controls because we are always concerned about contamination. And we never see any similar phenotypes in our negative controls. So I don't know if that helps explain it, but.
Yeah, a question from Judith Bronstein. Fascinating. Can you share more about how you will study coevolution with these techniques?
Well, one of the things that I'd really like to do is think about some of the issues of compatibility between the modern and contemporary genotypes. So doing some combinations. One of the problems that we've had is we don't have a very wide range of years, and there's a lot of heterogeneity in the herbarium specimens that we use. So if we can get a finer resolution of years, I think, in collections across the landscape, I think we can really start to look at issues of compatibility, especially with external environmental changes such as like nitrogen deposition. So once we have the methods further developed, I think there's some potential there, but we're going to need to have a little more luck with getting a little bit further back in time in order to start asking some of these questions.
Well, thank you, Renee. That was a great talk. Moving on to our next speaker. Next, we've got Priyanka Gera, who's going to talk about diversification and evolution of chitinase 1 in neotropical frogs, an enzyme at the forefront of the battle against BD infection.
Thank you so much, Freya. Hi, everyone. Thank you so much for coming today. My name is Priyanka Gera. We can all hear me, right? Okay, perfect. Thank you. My name is Priyanka Gera. I'm an undergraduate at St. John's University working in Dr. Santos's lab. And today I'm going to be discussing the diversification of chitinases in neotropical frogs and their role against the fungal infection caused by Batrachochytrium dendrobatitis. So since I submitted my abstract to this conference, we decided to expand my project a little more and not focus on just one chitinase gene, but rather multiple. So I'll be talking a little bit about that today. So first off, what are chitinases? Chitinase is an enzyme that is responsible for hydrolyzing chitin, which is a polymer made up of beta-1,4 linked sugar residues. It is an integral component to fungal cell walls as well. As we can see in the diagram at the top left, it shows the components of a fungal cell wall, and chitin is shown in this blue boomerang-like figure, and we can see that it forms about 50% of the entire fungal cell wall. The structure of the chitin monomer is shown in the diagram in the bottom left here, and what chitinase does is it comes in and it breaks down the glycosidic bonds that form between these chitin monomers. So what is BD? BD is the abbreviation for the fungus Batrachochytrium dendrobatitis. It causes the skin disease in amphibians, chytridiomycosis, and it's otherwise known as the silent killer fungus. It has ravaged through the world's amphibian population since the 1970s, and it originated in Asia, but was spread globally by humans via trade. What happens is, in this disease, the chytrid fungal spore ends up burrowing itself in the amphibian skin, developing into a thallus, and ends up maturing and then releasing more spores into the environment, further infecting the amphibian's own skin, but also infecting other amphibians in the vicinity. Chytridiomycosis has threatened many species with extinction, but some species are resistant, including Xenopus flavus, Rhina cataspina, and Bufo hipponicus. But the question remains, why are these species resistant? What do they have that the other species don't? And so this is where we believe chitinases might come into the picture. Because BD is a fungus, part of its cell wall is made up of chitin, and chitinases break down the chitin. And so these enzymes may play a role in the innate immune response against this fungal infection in resistant amphibians. So kinases are glycoside hydrolases because they break down glycosidic bonds. And we can classify them into two well-known families, glycoside hydrolase family 18 and family 19 as well. Family 19 mostly consists of plant kinases though, so we're going to be focusing on family 18, which includes kinases for animals and fungi as well. One of the main differences between these families is the structure of the catalytic domain of the enzymes. Another way we can classify kinases is based on their activity. Are they active or are they not? If they are active, then these enzymes can break down chitin, and within their amino acid sequence, we'll find this conserved motif at residue 135, DXX, DX, DXE. D represents aspartic acid, X represents any other amino acid, and E is glutamic acid. And aspartic acid and glutamic acid are crucial for the glycohydrolytic activity of these active enzymes. Now, some kinases are not active, so they can't break down chitin, and so they're referred to as chitinase-like proteins, which is abbreviated by CLP. This diagram on the left sums up all the different types of kinases that have been identified in different organisms, specifically from family 18. We can see, for example, for mammals, there's 3 different groups. You've got the KEAT1 enzyme, AMCAS, and chitinase-like proteins. Insects have 11 different groups of chitinases, but amphibians are not on this list. And that's because the chitinases in amphibians have still yet to be identified and characterized. In my project, though, I'm focusing on 3 different chitinase genes that have been found in mammals, but we've also identified them in the amphibian genome. The first gene being KID1, the second KIA, and the third KEAT1. So just a little summary, a little background about all 3 of these genes. KID1 is otherwise known as stable and interacting chitinase-like protein, and it is not active, so it cannot break down chitin. It is also one of the more recently identified chitinases in humans back in 2006. And it was identified to be a ligand for the stabilin 1 receptor in macrophages and endothelial cells in the liver, the spleen, the bone marrow, and lymph nodes also. KIA is the second gene that I'm studying. Its enzyme is known as acidic chitinase. It is an active enzyme, so it does break down chitin, and it was first identified in humans back in 2001. Now, this is an interesting gene because it has a dual function. This gene was found to be active in the lungs of humans and also in the GI tract. So it has a dual role in immunity, especially as it relates to asthma, but also in digesting chitin-containing particles that the organism might consume. KEAT1 is the third gene that I'm studying. Its enzyme is known as ketotriosidase. It is an active enzyme, so it does break down chitin, and it was first identified in humans back in 1998. And this gene also has a function in immunity. It is expressed in neurodegenerative diseases, lung and liver diseases, and also has antifungal activity. So chitinases have been previously identified in different organisms, humans, mice, primates, and zebrafish. Specifically in humans, there's 6 chitinases that we know of, 2 of which are active, being KEAT1 and KIA, and there are 4 chitinase-like proteins that have been identified. Specifically in zebrafish, during their embryo development at different stages and in different amounts, 6 different variants of KIA have been identified. Specifically, KIA3 was characterized to have a role in digestion, as well as to have antifungal activity, which further provides evidence for this dual role of KIA. In amphibians, however, so far only KIA has been found. Back in 2002, the toad pancreatic chitinase was the first chitinase to be identified in amphibians, and later that year, gut chitinase was also identified in the genus, in the species Xenopus flavus and also in Ranacattaspina. So I have been able to identify the presence of KID1 and KIA in the amphibian genome, and we're currently working on gathering and analyzing data for this third gene, KEAT1. So this entire methodology was a team effort, and it's quite complex, so I'm going to try to briefly summarize it into 4 different modules. So we used 2 different sets of data. For 41 different species, we used our own unpublished data, 75 million pair and 150 base pair reads. And we also used, for about 35 different species, data from the NCBI Sequence Read Archive public database. So first, starting off with our own unpublished data, we cleaned our reads using a multi-assembler de novo transcriptomic workflow. We used Busco as our quality control, And we annotated our reads as well using BLASTX and the UniProt library. After that, we isolated the genes that we were studying. So we've done this so far for KID1 and KIA. Next, from the genes that we isolated, we found a reference gene, so the most complete gene that we could find with a proper start and stop codon. We also ran it through BLAST just to once again confirm its identity and make sure it was the chitinase. At that point, we harvested additional sequences from NCBI. We harvested annotated cDNA sequences and also used sequences we found from the Transcriptome Shotgun Assembly database. We also standardized the names of all the genes that we harvested just so it'd be easier for us to identify them. We aligned all the sequences that we had harvested using Decipher, and we further cleaned our alignments by truncating any repetitions or errors that we found at the ends of the sequences just to get a more complete gene. And we also created a phylogenetic tree using tree estimation and bootstrap support. Another way we further cleaned up the sequences that we had was by removing any gaps as long as they were present in more than 80% of the taxa that we were studying. And we used the Amphibia chronogram published back in 2014 to help with the Hi-Fi analysis preparation. So in module 4, we ran parallelized Hi-Fi runs to test and find evidence for positive selection on either individual amino acid sites or on the branches of the phylogenetic tree. And we did not use a priori selection. In other words, we did not impose a hypothesis. It was a straightforward analysis of positive selection. And within this Hi-Fi package, we ran 5 different programs: Abstral, Busted, Fel, Foobar, and Meme. Now for Abstral, Busted, Fel, and Meme, we set the p-value at 0.01, but for Foobar, which has a different algorithm, the Bayesian posterior probability was cut off at greater than 0.91. So I'm going to be sharing the results today on 2 of the 3 genes that we're studying, KID1 and KIA. So looking at KID1 first, this is the phylogenetic tree that we produced. The green represents all the frog species, the orange being the salamanders, and the blue being the caecilians. So a total of 77 different species had the KID1 gene present, of which 70 were frogs, 3 caecilians, 4 salamanders. Now, of the 70 frogs, 55 are neotropical frogs. Neotropical just referring to the geographic location as to where the frogs are found, so South America and Central America. And this gene was 397 codons long. Looking at our analysis for positive selection, of our 5 tests, only 1 of them showed some evidence for positive selection. So Abstral, Busted, Foobar, and Fel did not show any evidence. In fact, as we can see in this diagram below, for Fel, it showed overwhelming evidence for strong negative selection throughout the entire KID1 gene. Meme is the one that did show some sites that are evolving under positive selection with a p-value less than 0.05, but of all of these sites, only 7 of them are strongly significant with a p-value less than 0.01. For the KIA gene that we analyzed, we actually found 5 different variants within our data set. So looking at the first variant that we identified, KIA A, we found it in 66 different species, 60 of which are frogs, 2 caecilians, 4 salamanders, and of the 60 frogs, 52 are neotropical frogs. And this gene was 540 codons long. So for our tests for positive selection, some of our tests that we ran did show evidence for positive selection. So looking at Abstral first, it showed evidence in about 7 different branches of the phylogenetic tree, including the genus Bombina, the genus Pelobates, and also within the Ranidae family. Busted also did show evidence for positive selection at about 3% of the sites. Foobar and Fel did not show evidence of positive selection. In fact, again, we can see for Fel in the diagram below, it shows mostly negative selection throughout the KIA A gene. Meme showed quite a few sites that are evolving under positive selection with a p-value less than 0.05, but of all of these sites, only 8 of them are strongly significant with a p-value less than 0.01. Looking at the second variant that we identified, which we labeled KIA B, we found it in 25 different species, of which 21 are frogs, 1 salamander, and 3 caecilians. And this gene was also the same length as KIA A, 540 codons long. So for our tests of positive selection, some of the tests that we ran did show evidence. Absol, for example, showed evidence of positive selection in 3 different branches of the phylogenetic tree, specifically for the genus Nanorena and within the Branidae family as well. Busted also showed evidence of positive selection at 2% of the sites, and Fubar showed evidence as well at 1 site. Fel, however, did not show evidence. In fact, as we can see from the diagram, it showed an overwhelming evidence for strong negative selection throughout the entire Kia B gene. Now, Meme did show quite a few sites evolving under positive selection, but of all of these sites, only 6 of them are strongly significant with a p-value less than 0.01. Looking at Kia C, the 3rd variant that we identified within our data set, 25 different species had Kia C, of which 21 are frogs, 1 salamander, and 3 caecilians, so quite similar to Kia B. And just like Kia A and Kia B, Kia C was also 540 codons long. For our turmeric tests for positive selection, some of the tests that we ran did show evidence for positive selection. So Absol showed it specifically at 4 different branches within the phylogenetic tree, including that for the genus Microhyla and the genus Rhacophorus. Busted also did show evidence of positive selection at about 2.5% of the sites. Fubar and Fel, however, did not show any evidence for positive selection, and we can see in the diagram below, once again, Fel showed overwhelming evidence for strong negative selection throughout the entire Kia C gene. Meme, once again, did show quite a few sites that are evolving under positive selection with a p-value less than 0.05. But of all of these sites, only 2 of them are strongly significant with a p-value less than 0.01. So looking at the last 2 variants that we identified, Kia D and Kia E, we only found them within a really small portion of our data set. So for Kia D, we only found it in 3 different species of caecilians. And for Kia E, we only found it in 3 different species of frogs, specifically 2 from Bombina and 1 Pixie-cephalus. And unfortunately, because we only found them in 3 different species each, this was not enough data for us to follow through with a positive selection analysis. However, what we can say is that the sequences for Kia D and Kia E were distinct from A, B, and C. And so they are distinct and different sequences. So we did find 5 different ones, but in terms of an analysis and any further analysis, we won't be able to do that as of now with this small amount of data we found it in. So the fact that we found 5 different Kia variants, though, within our data sets is congruent with the fact that the amphibian diet consists of chitin-containing organisms, specifically insects with a chitin exoskeleton, and they would need chitinase in order to digest the insects that they're consuming. And of the chitinases that I spoke about today, Kia is the only one with that dual function in digestion as well. We also found an interesting link between Kia and BD. So most of the data that we used was skin transcriptomes. And the fact that we found 5 different variants within the skin transcriptomes suggests that perhaps Kia is the enzyme that plays a role in hydrolyzing the BD spores that embed themselves in the amphibian endothelial cells. So we used data from frogs, salamanders, and caecilians. For the caecilians, we used data partial and complete genomes that were available. We had 90% of our data was skin transcriptomes, 10% were other tissue types. We overall concluded that the KID1 gene is not under positive selection, and so it's mostly conserved throughout the amphibian species. Kia A, B, and C do have sites under positive selection, so those sites are evolving much faster than the other sites. And unfortunately for Kia D and E, we don't have enough data to continue with analysis for that. And in regard to the 3rd gene that I spoke about today, KIT1, we're currently working on gathering and analyzing more data for that. So with that, I'd like to acknowledge my lab members, my co-authors, collaborators, and also financial support that I received from St. John's University, NSF grant, as well as MSU. And with that, I take any questions. Thank you so much.
Thank you so much, Priyanka. We have time for 1 question. Yeah, Sarah McPeak asks, nice talk, Priyanka. Is there any work linking presence of these chitinase genes with historical prevalence of BD infection in the species expressing these genes? That is, can we identify BD as a potential agent of selection on chitinases?
So we actually don't have any information on that. In fact, even just within the amphibian species, it's hard. We don't have enough information to tell which species are susceptible to BD and not. And so not a lot of work has been done to see if chitinases are actually linked with the BD infection. So that's something that we're hoping to work on. And the next step in the project would be to determine that and see whether or not these genes, the ones that showed evidence for positive selection in those particular branches, are they susceptible to BD or not? So that's actually something we're working towards. We don't know that as of now, if they're linked or not.
Thanks a lot, Priyanka.
Thank you.
All right, thank you, Priyanka. Next up, we've got Maria Rebolleda-Gomez talking about evolution of metabolic interactions and a long-term community evolution experiment.
Yeah, thank you, and thanks everyone for coming to my talk. Let's see there. So before I start, I just want to take a brief moment to recognize that these have been really hard times. For everyone. Like, I've heard some people saying that they've had trouble finishing the talks and things like that because of the pandemic. There's been a lot of violence, and none of these things have affected us all in the same way. And so I just want to recognize all that and thank you especially for being here in these times. And so this past year has really highlighted I think the importance of interdependencies and interactions, and the cost of rendering those interdependencies invisible. And so an example is, like, much of our food gets produced in crowded spaces by people with little or no access to healthcare, and we don't tend to see all these things, but these workers make our lives possible. And so in a similar way, I also want to thank all the people that have made it possible for me to go to the lab during the pandemic, the janitors, all the administrative stuff, and all the people that have put their life at risk for us to keep the labs open. In addition, this work is a big collaboration. It's a collaborative effort between many people. So I want to thank all the co-authors on this work. Sylvie Strela, Jackie Folmar, which is an undergrad extraordinaire, Georgie Badzik, Alicia Sanchez Gorostiaga, Nancy Liu, and Alvaro Sanchez. And I especially want to acknowledge the immense contribution of Sylvie to this work. And so, as I said before, I'm more generally interested in interdependencies and thinking about how organisms affect each other and their shared environments. And this is, I think, particularly important in the case of microbes, because microbes live in very complex communities where they interact with each other through a lot of signals, enzymes, antibiotics, and a lot of metabolic diffusible compounds that are constantly modifying their environment. And so in these communities, the ecology affects evolution, and evolutionary change can feedback and affect the ecology. And this makes really hard to predict evolution, and it would make it, it seems to me that it could increase the role of historical contingency, because if you have a mutation that has an effect on the ecology, this ecology can then amplify the effect of this mutation. And so that way it can create sort of alternative paths. But most of the experiments we've done in microbial experimental evolution have been with a single strain or a couple of species, and so it's really hard to see how that affects historical contingency. On the other hand, a lot of these interactions between bacteria are mediated by metabolism. And it's tuning the metabolism is easier to evolve than complete new metabolic structure. And so metabolic constraints could in some way increase the predictability of ecology and evolution. What I'm arguing here is that, you know, you cannot completely change metabolism that much in a few generations, so that at least in the short term, these interactions are bounded by the possibilities of the metabolism itself. And so to test these ideas, I'm taking advantage of previous work in the Sanchez lab, showing that when you isolate microbes from very different sources, when you get a diverse pool, and then grow them in a very simple environment with a single sugar as the only carbon source, then communities converge, at least at the family level. So this is the case for glucose, but the same happens for all carbon sources that we've tested. And what you get is these similar functional guilds, where you get not only the same families represented, but the same families more or less in the same proportions. And so we get Aromonadaceae or Enterobacteriaceae, which are more closely related to each other, and they're fermenters, and I'll talk about this. So basically, in the presence of oxygen, they can ferment carbon sources. And we also get Pseudomonadaceae and Moraxulaceae, which are also more related to each other than they are to the other two families. And these ones are respirators, so they cannot ferment in aerobic conditions, and they actually cannot ferment sugars. And so this structure happens because fermenters can grow faster in glucose. So initially they store, so I'll talk a little bit more about this in detail, but basically fermentation is faster, or is the strategy that microbes tend to use when they're growing really fast, but it's not the most efficient. And so as microbes grow faster in glucose, as I said, this is not the most efficient growth. And so they consume the glucose, but they also produce a lot of organic acids as byproducts. And then the respirators are better at consuming these organic acids. And so they take advantage and they can grow at this point. And so the question here is if we can use simple metabolic rules to predict evolutionary change in these simple communities. And we know from work with E. coli, and this now we know applies for different fermenters, the relationship between growth rate and organic acid production rate is very defined. And so when you have very slow growth rates, E. coli or all these fermenters switch to a respiration metabolism, and they don't produce any waste product, organic acid waste product. But after a certain breakpoint, they actually, as growth rate increases, they become more and more wasteful and start producing more organic acids. And this is a constraint of metabolism because it seems from what we know so far that it has to do with the amount of protein available for multiple functions in the cell. And so cells need to allocate the protein in different ways. And fermentation is much cheaper in terms of number of proteins than respiration. And so if you are using a lot of your proteins to grow, you don't use them in metabolism. And so we can imagine that after selection in a single carbon source, we might get that fermenters then evolve a faster growth rate. But as they do, they would evolve increased organic acid production rates. And so we would get an increase on a respirator to fermenter ratio. Now, it is also possible, but it seems less likely because it would require a larger restructuring of the metabolism, that you could instead actually push that trade-off forward, that Pareto front, and get increased growth rate while at the same time you're doing a more efficient metabolism and you're not producing that much waste product. Now, of course, I know that also respirators could evolve in response and affect this respirator to fermenter ratio. But in interest of time, in this talk, I'm only going to address the fermenter side. And so to evaluate these ideas, we perform an experiment similar to the community assembly ones I just described. So we grab a diverse pool of microbes from soil, let it stabilize for 9 transfers in glucose. And then from this stabilized community, we started 12 replicates and transferred them again every 48 hours for approximately a year, or 826 generations of bacteria. And afterwards, we isolated different colonies, sequenced the isolates, and we have found that these communities are mainly comprised of 3 big players: Aromonas and Klebsiella, which are both fermenters, and Acinetobacter, which is the respirator in this case. And so when we measure the growth and their organic acid production, what we see is that there is a relationship, and the faster you grow, the more wasteful you are as well. And so, but slow growing strains evolve and really quickly and increase growth rate. And as they do, they also become even more wasteful. Instead, the fast growing strains, the Aromonas, they started growing really, really fast on glucose. And as they evolved, they actually produce a little bit less organic acid to the sort of detriment of their growth rate, only slightly. And we currently have more data here because, as you can see, the error bars here are huge. This was preliminary data, but I haven't finished analyzing the growth rates. But what was puzzling for me is that Aromonas is the most common bacteria throughout the experiment. And so how could it kind of persist and kept evolving, but actually become less good at growing? And at least part of the explanation is that Aromonas actually evolved a decrease in lag time. So now every transfer, Aromonas could start growing earlier, taking advantage initially of the glucose available. And we think that that might be instead the strategy. And so from this part, I think I have shown some data that supports the idea that evolution is constrained by metabolism. And so slower growing strains evolve increased growth rate, but do produce more organic acids as a byproduct. And growth rates of rapid growing strains did not evolve. And we think instead these strains evolve shorter lag phases. And this might be because once you're growing really fast on a particular resource, it might be very hard to become even better. And so there might be other sort of less constrained avenues of evolution. But how does evolutionary change affected the community composition? And one thing that was amazing, so I recently we sequenced all these communities, we got the 16S, and so we can look at the abundance. And what was crazy to me is how convergent the ecology was over time. And so here are the 12 replicate lines, and over time, and it's the ratio of respirators to fermenters in the y-axis. And as you can see, yes, there's a point where one population gets tons of respirators, but not only the lines stay more or less together, but they follow the similar pattern where we first get an increase in the respirator to fermenter ratio, then these decreases, then there's a second bump, and then decreases. And when I see the like bar plots of these, it looks even crazier. You can really see that it's not only the RF ratio, but even the relative proportions of the different taxa in these communities that is changing more or less synchronously. And so what we don't know here, one thing is if the spikes in RF ratio were due to some external environmental stress, like one day for some reason, the incubator was slightly different or whatever else, or if they had to do with internal changes, so mutations and phenotypic changes that are happening, and we know that are happening within the communities that might create some of these changes. And what I think is really cool is that it seems like no matter if it's external or internal, the relationship between these data, it seems like we're maintaining a ratio more or less stable despite these perturbations. And at the beginning and end points have a very similar ratio. So it seems to be some buffering going on. And so at the beginning, when I got this data, I was like, okay, this cannot be real. Maybe this is just an artifact of sequencing. And so I plotted the RF ratio calculated with plating against the RF ratio calculated with the Chitinase. And it does seem like there is a really good correlation. There is a slight bias of Chitinase that favors respirators, and this is a fairly well known bias in Chitinase. It favors Pseudomonas and related taxa. And so as I was saying, the RF ratio is maintained overall, but there are clear perturbations. And while we don't know if those perturbations are internal or external to the system, they happen more or less at the same time in all of the populations. And we have some samples that we have taken of isolates between those different time points. And so we can look more closely at what's going on. And one possibility of internal change would be that initially, at the first increase of the ratio, what we have at that point is that the Enterobacter, the Klebsiella, has evolved a higher secretion of lactate, as it evolved a higher growth rate. And so that might provide nutrients for the Acinetobacter to then grow. And so that could explain that first peak. Then the Aeromonas evolve a shorter lag phase. And so that might create this slight decrease in acetate production, which would again shift the ratio towards the fermenter side. But then at a later time point, both the Enterobacter and the Aeromonas evolve again higher production, but this time of acetate. And this might relate to, again, an increase in the respirator. So there's a possibility that this is actually has to do with a high convergence of the phenotypic changes of each of the taxa that then actually translate to high convergence at the community level. And so what I can conclude from this talk is that phenotypic evolution and community composition are highly repeatable, at least in the short term. And this is important because in the long term, I do expect that there might be some more possibilities of rearrange metabolism and break some of these sort of weak constraints. And that might affect the ecology in an unpredictable way. But despite perturbations, which I said I'm not completely sure if they're internal or external to the system, the RF ratio remains more or less constant, and it seems to be buffering, which I think is really cool. So it does seem like we can use simple metabolic rules and constraints to predict evolutionary change, at least in simple communities and in the short time scale. And with that, I would like to thank everyone at the Sanchez Lab, my funding from the Yale Institute of Biospheric Studies, and a lot of people that make this possible. And I'll take questions.
Thank you, Maria. We have time for 1 or 2 questions. Bob Week asks, fascinating talk and beautiful slides. If you continued the experiment indefinitely, do you think you would see oscillation in the RF ratio?
Yeah, and we're starting to get into the respirators side of thing, because I also think an important part to what might be happening in these oscillations is an evolution also of the respirators to be more efficient at consuming the organic acids. I don't know if we'll keep seeing those oscillations. It is pretty striking, this ratio of respirators to fermenters also evolved in the Lenski experiment. So in the Lenski experiment, it's only E. coli at the beginning, but E. coli evolves these sort of acetate specialists and these glucose specialists, and they cross-feed each other. And they seem to get around this sort of 0.3 ratio, more or less. And so I don't know if that's what's going on with that particular ratio, but it does seem to be something relevant there.
Thank you.
Thank you, Maria. Okay, up next we've got Joshua Fowler, who's going to talk about grass-fungal endophytes in time and space. And then Joshua, I don't know if you're talking, but we can't hear you if you are.
Oh, can you hear me? Okay, cool. All right, hi, my name is Josh Fowler, and today I'll be talking with you all about using museum specimens to assess the response of grass-fungal endophytes to climate change. And this is work that I've been doing along with two undergraduate researchers, Ella Segal and Lainey Dufresne, along with my PhD advisor, Tom Miller at Rice University. And I want to start just with the observation that microbial symbioses are ubiquitous across the tree of life and around the world. We can observe them everywhere in nature. These are intimate interactions where microorganisms live inside of their hosts, and they can have important consequences for organisms' reproduction, growth, survival, and defense. Species interactions such as these form the biodiversity that we observe in nature. And many of them are mutualistic, where the microbial partner benefits its hosts and in the process ensures its own survival. And so here we can see some examples, which all happen to be mutualisms, including Wolbachia inside of bacteria, Symbiodiniaceae that live inside of corals, and legumes that live inside, Rhizobia that live inside of legumes, similar to what Renee talked about earlier. And while these interactions are commonly mutualistic, they are also often context dependent, where the outcome of the interaction depends on environmental conditions. And that means that the costs and benefits of the interaction may shift depending on environmental drivers. And so it's important to know how mutualisms and microbial symbioses in particular will respond to climate change. And it's a major question of, will mutualisms break down under climate change? And here on the left, we can see over the last century across the US, the change in mean annual temperature, and on the right, change in annual precipitation for the last century. And climate change involves heterogeneous changes in climate across space and over long time periods. Understanding how species interactions have responded to past climate change can inform how species interactions may respond to future climate change. But climate change occurs over periods longer than typical ecological research projects. And we don't really know how most microbial symbioses respond to environmental drivers today and how they have responded in the past. And there are two ways that species interactions can respond to climate change. The prevalence of interactions can change, and that could be, for example, a pollinator that declines in prevalence and can no longer provide the services that it was providing, or the outcome of interactions could change, where the pollinator may provide better service under future climate conditions. And it's also worth noting that vertical transmission in microbial symbionts can actually create a positive feedback between outcome and prevalence. Because the symbionts live inside of their hosts, they can affect their own prevalence within the population. And all of the examples that I showed earlier are actually vertically transmitted microbial symbioses. And these mutualisms may respond to climate change by providing resilience or breaking down. And so if a species interaction provides resilience under climate change, you might see that it would increase in prevalence and provide stronger, better outcomes for its partner, or it may break down and be hindered by climate change, where it declines in prevalence and provides worse outcomes of the interaction. And today I'll be focusing on epicloe fungal endophytes, which are widespread symbionts of cool season grasses. And here you can see the fungal endophyte, which is this hyphae growing inside the seeds of a squashed seed, pointed to in red. And these are facultative symbionts of many species of cool season grasses that are vertically transmitted. And that means that they grow inside of their host and then grow and reproduce asexually inside the host up into the seeds of the grass and are transmitted to the next generation. And these interactions, because they're vertically transmitted, are expected to be mutualistic. And in fact, they're known to provide drought and anti-herbivory benefits to their hosts. But these benefits are oftentimes context dependent. And so it's unclear how they will respond to different environmental drivers and how the benefits and prevalence of the symbionts may change with the environment. Some of the early work with fungal endophytes actually used herbarium specimens to study the taxonomic distribution of endophytes and found that endophytes are common across many genera of cool season grasses. And so hold that in your mind as we ask, how can we study the long-term dynamics of fungal endophytes? Well, natural history collections preserve specimens from many different species that are collected over long timescales and across broad spatial scales. And they've mostly been used to study taxonomy and biodiversity in the past, but they can also preserve evidence of ecological interactions. And we saw a really cool example of that earlier from Renee, talking about actually using genomics, but they've also been used to study phenology, pollination, and herbivory across time and across space recently in a bunch of cool and exciting papers. And so today I'll be asking, how have fungal endophytes responded to climate change? And in particular, we can look at how the prevalence of fungal endophytes has changed and how the outcome of the interactions have changed. And I'll break that down into slightly smaller questions by asking, how does endophyte prevalence vary across spatial environmental gradients in space? And then how has endophyte prevalence changed over time? And we can ask the same questions, how has the outcome of the interaction varied across spatial environmental gradients? And how has that outcome changed over time? And to answer these questions, we visited 4 Texas herbaria, including the herbaria at the University of Texas at Austin, Texas A&M University, the Botanical Research Institute of Texas, which is located in Dallas, and then Mercer Botanic Gardens, local Houston herbaria. And we'll be focusing on 2 grass hosts, Elymus virginicus, shown on the left in blue, and Agrostis hyemalis, shown on the right in green. Both of these grasses are commonly collected and have broad eastern US distributions. Both of their range edges overlap and occur approximately in the center of the US, and so they cover pretty much the entire eastern US. But we see that Elymus virginicus is a little bit more broadly distributed, at least amongst the collections. And here, these maps are showing county collection records of each species. And we collected 5 to 10 seeds from 804 specimens, and of which 535 specimens came from Agrostis hyemalis, and 269 specimens came from Elymus virginicus. And in collecting these, I visited the herbaria and looked under the microscope and looked for plants that had numerous seeds and weren't in flower. So this picture on the left is actually showing an Agrostis hyemalis flower, which I think grass flowers are generally underappreciated, so I wanted to include that here. But this is not a specimen that we could have collected from because it doesn't have seeds, although oftentimes an individual specimen might have some spikelets that were flowering and some that were already developed into seed. And seeds from each specimen were scored using microscopy. So for example, this Agrostis hyemalis specimen, which is seen here, we can take seeds from that and squish them under the microscope and look, and you can see fungal endophytes preserved just basically by chance that we can look at this. And it's a really amazing opportunity to see the hyphae of this fungal endophyte from this historic specimen and see the interaction from the past. And in addition, each specimen is linked to collection dates and locations. So this specimen was collected near Clark County, Georgia, all the way back from 1930, so over 90 years ago. And to assess the interaction outcome, we collected measurements of reproductive output from the images of herbarium specimens. And this part of this project was really driven by my two undergraduate co-authors, Ella and Lainey, who worked over the summer remotely as REU students with herbarium image data. And so for this Elymus virginicus specimen, we can count that there are 4 inflorescences, which are the flower producing structures. And we can also measure the length of each inflorescence. And so this specimen has an average length of 5.4 centimeters. And the length of inflorescence is correlated to the number of seeds that plants produce. So longer inflorescence length means more seeds. And so our collections are concentrated in the South, but they actually cover pretty much the entire Eastern US. And here I'm showing our collections for Elymus virginicus as triangles and Agrostis as circles. And as if you recall earlier, I pointed out that the rangeage of these species occurs in the middle of the US, and that actually coincides with the longitudinal precipitation gradient. And so I'll be focusing on longitude in this talk, basically to ask how does environmental variation change the interaction, but there's still obviously more complexity across latitude and through time that I won't necessarily focus on in this talk. In addition, our specimens are concentrated after about 1940, but they cover the entire time period between 1864 and 2016. To answer our first question, how does endophyte prevalence vary across longitude? We'll be looking at mean endophyte prevalence on the y-axis and longitude on the x-axis, where west is to the left and east is to the right. And according to that precipitation gradient, we would think that west is drier and to the east is wetter conditions. And each specimen is given a score 0 or 1 based on the seeds that we collected for symbiont status, where 1 means that the specimen is infected and 0 means that the specimen is not infected. And then we can calculate mean endophyte prevalence across longitude. And here the points show the binned means across longitude of our specimens. And the line shows the estimated mean prevalence from a generalized linear model. And this is kind of surprising, but we see that endophyte prevalence is actually lower towards the west in Agrostis hyemalis, which is the drier portion, when in generally we would have expected there to be high prevalence under drier conditions if we were expecting this fungal endophyte to provide drought benefits. And this actually reflects patterns that we've seen in contemporary population surveys that have been done by our lab. And so on the right, we see a similar decreasing trend towards the left, towards the west. And these contemporary surveys involve driving around and visiting populations and collecting multiple plants from the population and scoring seeds from those populations. And this covers a much smaller longitudinal gradient because it involves actually going out and driving and collecting all within one season. But it's encouraging that we see a similar pattern across contemporary surveys and these herbarium surveys. And they both suggest that potentially Agrostis hyemalis is not providing benefits under drought conditions. However, there's not a clear longitudinal trend in Elymus virginicus. So here we see the specimens, and there's potentially, there's not really a significant trend across longitude, but contemporary surveys still don't have a very strong trend, but they suggest drought tolerance benefits with higher prevalence in drier conditions to the west. And in addition, we know that Elymus virginicus from previous work in greenhouses can provide drought tolerance benefits. And so together, these results can tell us that species may provide different benefits to their hosts under different environmental conditions, where Agrostis hyemalis may experience weaker benefits in drier conditions, and Elymus virginicus may experience slightly greater benefits in drier conditions. So now that we know maybe how environment may change the prevalence, we can ask, how has endophyte prevalence changed over time? And here we'll be looking at a very similar graph where we're looking at mean endophyte prevalence versus longitude, except we'll color our points by time, where samples from the year 1930 binned around the year 1930 are shown in gray, and samples binned around the year 2000 are shown in green. And we actually see that prevalence has increased over time in Agrostis hyemalis. So the curve across longitude has increased in prevalence. And we see a very, very slight decrease in Elemus virginicus across our samples, shown in blue on the right. And what might be driving these changes across time? We know that these plants might respond differently to environmental drivers. And overall, these populations have experienced an increase in precipitation. And so potentially this increase in precipitation is what's driving the changes in prevalence. And so we see that potentially Agrostis hyemalis is showing signs of resilience to climate change, where it has increased in prevalence, while Elemus virginicus is to a lesser degree declining in prevalence and potentially being disrupted by climate change. And now we can look at how the interaction outcome varies across longitude. So for this, I'm going to focus on Elemus virginicus, where we have 92 specimens with both endophyte scores and inflorescence measurements. And I'm focusing on Elemus virginicus because it has a little bit more compact inflorescence structure, which makes it easier for us to measure. And we have measurements for Agrostis hyemalis, but they're less clear since the specimens are more splayed out with many spikelets. And so first we want to know how do endophytes affect the number of inflorescences across longitude? And here we'll look at the number of inflorescences on the y-axis, across longitude on the x-axis, and we'll be filling endophyte status, filled points for endophyte status where plants are infected, and clear points where the specimens are not infected. And overall, we see that inflorescence count does vary across longitude, increasing in wetter environments, but it's not actually affected by endophyte status. And endophyte symbiosis slightly decreases inflorescence length, but it doesn't vary across longitude. And so there's not a clear effect of endophytes on interaction outcome, but we have limited specimens. But we can look, just for now at least, if these effects have changed over time. And there's not a clear endophyte effect on interaction outcome over time. Here we're looking at the number of inflorescences through time, and there's no trend and no effect of endophyte status. And similarly, there's not a strong trend through time, and endophytes have a non-significant but negative effect on inflorescence length. So overall, these specimens do show variation in reproduction, but there aren't clear trends in interaction outcome over time. So potentially there's been some negative effects or disruption in the outcome of this mutualism, which may be driving the changes in prevalence. And so just to sum up, herbarium collections reveal interaction resilience to climate change for Agrostis hyemalis and partial interaction breakdown for Elemus virginicus. And this is potentially driven by changes in precipitation across the southeastern US. And endophytes hosted by Elemus virginicus have slightly negative effects on reproduction, but there aren't clear temporal trends. And so in thinking about how microbial symbioses have responded to climate change, these results are really the first insight into the long-term dynamics of grass fungal endophytes and demonstrate both mutualism disruption and resilience. And environmental context dependence may drive large-scale spatial patterns and temporal patterns in the prevalence and outcome of microbial symbioses. And it really raises new questions, such as across the host range, how do differing magnitudes and directions of climate change contribute to endophyte responses? And we looked pretty much across the southern US, which has seen increases in precipitation. And so potentially these interactions may respond differently to different climate change drivers. And we can also ask, are there traits of hosts and or symbionts that may explain the resilience or disruption of interactions? And this would be interesting and a great opportunity to look across herbaria, which house many different species, and potentially could see if there are different species which are responding in similar ways. And with that, I just want to thank you all for your attention. I wanted to thank my co-authors, Ella, Lainey, and Tom, as well as the NSF and the Texas Ecolab grant program for funding, and all of the herbaria, which provided specimens and assistance, as well as other undergraduate researchers who helped with this project. And with that, I can take any questions.
Thanks a lot, Joshua. We have time for 1 or 2 questions. Kelly asks, do you think that drier conditions might affect whether the endophyte can successfully colonize seeds?
Yeah, so I think that's a really good question. And imperfect transmission, so where the endophytes actually don't make it into the seeds and don't germinate in the next generation, is a big thing that people have studied in fungal endophytes. And for the most part, there is in sort of other greenhouse experiments, there's not a huge amount of imperfect transmission, at least for Agrostis hyemalis. But I think it is important. It would be hard for us to look at because we've collected only 5 to 10 seeds from each specimen. So we can look at imperfect transmission at least a little bit, but kind of only in a very rough way, just because we have so few seeds to estimate the imperfect transmission.
The next question from Renee. Great talk, Joshua. Are endophytes harder to detect in older specimens? Also, a follow-up question. Do you know how herbivory would map onto the range distribution of this plant species?
Sorry, what was the second question?
The follow-up question was, do you know how herbivory would map onto the range distribution of the plant species?
Yeah, so for the first question, it seems that we haven't seen that it's harder to detect the endophytes. And for the most part, the looking under the microscope, the quality of the seeds when you squish them looks very similar between the preserved specimens and the new specimens. So it seems like it's preserved pretty well. Although obviously I imagine that it is slightly harder to see endophytes, but because we're looking at sort of the physically preserved endophyte as opposed to trying to sequence or culture them, I think that it's still there. And then if I had to guess, I've been thinking that potentially there's more herbivory to the east, just because it's potentially a more productive environment, but that's not something that I necessarily have a great sense of. But I do think that it would be interesting to think about how are other ecological interactions driving this as opposed to just climate change. Like, has there been an increase in herbivory, or have there been differences in plant competition or something like that?
Thanks a lot, Joshua.
Yeah, thank you.
So our next speaker, Chandra Moffat, has actually had to cancel. And so we're going to take about a 20-minute unplanned break until 5, but please return for Rita Grunberg, who will be at 5 p.m. All right, we can resume here. As I mentioned in the chat, our 4:40 canceled due to illness, but our 5 p.m. speaker is here. So without further ado, let's welcome Rita Grunberg, who's going to talk about infection history alters the trajectory of parasite community assembly.
Rita, are you there? Oh, there we go. Excellent.
Okay, can you hear me? Yes, awesome. Okay, move this little Zoom. Okay. Hi, everyone. My name's Rita, and I'm a postdoc in Charles Mitchell's lab. And today I'm going to talk about how the infection history of a host can alter the trajectory of parasite community assembly. And before I proceed, I want to thank my co-authors, Brooklyn and Charles, for being really great people to work with. So as many of us know, a host can harbor multiple parasite species. An example of this is shown here. Hold on, damn it. Trying to get, okay. An example of this is shown here, where we have an individual grass host that is co-infected by these 3 pathogens, here, here, and here. And co-infections can be really important for parasite community structure, not just because of what you're seeing here. It's because these parasites can interact potentially with one another within their host. So these parasites can interact either via competition for a shared host resource, or they might interact indirectly via the host immune response. And this example here of this grass and these fungal pathogens happens to be our study system of tall fescue and these 3 pathogens, which are Colototricum, Rhizoctonia, and Puccinia. And these 3 pathogens frequently co-infect their host. So since they frequently co-occur within their host, it's likely that these pathogens may interact. And so these interactions may depend on the spatial and temporal scale within their host. And also these interactions can be really important for among host transmission because these parasites can alter each other's growth rates within their host. So for example, if we have just these two pathogens, Coleototricum and Rhizoctonia, these two pathogens might interact negatively within their host, where this pathogen is negatively impacting the other pathogen's growth rate and vice versa. And then let's say we have a host that's infected with just Rhizoctonia and Puccinia, where maybe Puccinia is having a positive effect on this pathogen's growth rate, and this parasite is having a negative effect on this parasite's growth rate. And so parasites don't just interact with each other, they are part of an entire microbial community within their host. So these parasites might interact with other members of the community. And another important member in this community is epichloi, which is a fungal endophyte, which we just heard a talk about. And so epichloi also has the potential to interact with these three pathogens. And so they might interact directly. And then furthermore, this can modify interactions among the other parasite species. So you can have these complex interaction networks going on within an individual host. And these interactions within this host can have implications for parasite community structure because, as I said before, they're potentially altering each other's abundances within their host. And so what we wanted to know was whether the infection history can alter the trajectory of parasite communities. So specifically, if we have a host that's infected with one pathogen, is it going to lead to a different parasite community, whether it's infected with a different pathogen or no pathogens? And so to answer this question, we did a field experiment. And before I start this field experiment, I want to thank Brooklyn again for all of her hard work in leading this field experiment. And so what we did here was we inoculated plants in the lab, and we inoculated these lab-raised plants with two fungal parasites, Coleototricum and Rhizoctonia. And then we also inoculated them with epichloi, which is that fungal endophyte, which is proposed to be a mutualist in this system. So we inoculated these plants with these three symbionts in a full factorial design. And over here is a picture of the Coleototricum culture that we use for a source of inoculum for infections. And then over here is another fungal culture of Rhizoctonia, which we use for the source of inoculum for Rhizoctonia infections. And then to manipulate epichloi presence, we use different seeds because these are transferred by seeds. And so after these plants were inoculated with the symbionts, we kept them in a dew chamber for a little bit, which is shown over here. And then we deployed these plants in the field. So after we inoculated them in the lab, we put them in the field, and here is a picture of one of those plants. So here's a plant that was inoculated in the lab, and you can see it's kept in this little container over here. And then these plants were deployed and placed within this little arena over here. And these flags represent each one of these flags represents an outplant. And so the idea here is one of these plants are going to get future infections from these resident plants within this arena. So this is our source of future infections. So after we place these plants in the field, we track their infections. And so here you can see a picture of two field technicians, Julia and Ryan, and they are surveying the plants for disease. And so they surveyed these plants for disease for about 2 months. And so just to give you an idea of what was going on in terms of in the background, what epidemics were going on and what parasites they could obtain in the field, I'm just going to show you some long-term infection data that we have. So here are the 2019 parasite epidemics from a long-term field survey we've been conducting in the lab. And so here on the y-axis is prevalence, which is the proportion of plants and leaves that are infected with a parasite. And then on the x-axis is time, so we did monthly surveys starting from January to December, and this has been going on for several years. And so here are our three main fungal parasites, Coleototricum, Rhizoctonia, and Puccinia. And just to give you an idea of when these surveys are happening and what potential parasites these plants can get, here in this gray box, this shows when the survey was happening. So the surveys happened from September to October. And the important thing to note here is that these plants can get infected with all three parasites because all three parasites are in a pretty high prevalence within the field. And so after we did these surveys, we mapped out the parasite communities of these plants. So we mapped these parasite communities in multivariate space, but just to tell you what parasites we're working with, we are working with the same 3 parasites that I discussed in the beginning, Colletotricum, Rhizoctonia, and Puccinia. And then we also have a new parasite in our system, which is gray leaf spot. So we mapped these parasite communities in multivariate space, and we specifically did an ordination of these parasite communities of all host individuals across the entire survey. And so here's an example of one of those community patterns that one of the mapping of the parasite community. And so here is just a subset of the data just to orient you as to what's going on. So here's an NMDS plot of all the plants that were within this treatment group. So as I mentioned before, this was a full factorial design. So we have all combinations of all symbionts, of the 3 symbionts that we use. So here are plants that were inoculated with Epicloe, Colletotricum, and Rhizoctonia. So the fungal endophyte and then the 2 pathogens. And so here I just showed you, I'm showing you where the parasites map out in multivariate space. So here we have gray leaf spot, down here we have Rhizoctonia, over here we have Puccinia, and then over here we have Colletotricum. And so to generate this ordination, I used parasite severity data within this community matrix. And then also what you might notice is there's this random dummy variable or thing called dummy in the middle. And so what I did is I coded a dummy variable into the community matrix to include uninfected hosts. So all hosts will start from the same community state, which is uninfected. And so as I mentioned, this is a full factorial design. So now that I've oriented you with one treatment group, I'm going to show you all the data that we have. So here are the parasite community patterns across all the treatments. So these first 4 panels up here represent all the plants that were supposed to be that were not infected with Epicloe or not inoculated with Epicloe. And then down here are plants that are supposed to be inoculated with Epicloe. So these are the Epicloe positive seeds. And then we also have, if you look over here, these are the plants that were infected with Colletotricum in addition. And then these were co-infected with the pathogens, and then these were just infected with Rhizoctonia. And so as you can see, there's just generally differences in the spread of the data, but we're going to actually analyze this later, and I will show you some analyses later. But this is just to show you the general, all the data. And so the first question I wanted to ask was whether these communities varied in terms of time and how much they varied based off of the inoculation treatments. So first, I looked at temporal variation in parasite community structure. So here's just an example of what I mean by that. So here is just the general pathway of a parasite community from an individual plant. So this is plant ID 88. And you can see how much it travels over time. And so a way in which I measure temporal variation in parasite community is by measuring the length of these vectors. So I measured the Euclidean distance that these communities moved over time. And another way of looking at this is this graphic over here. On the x-axis is the time, and on the y-axis is the Euclidean distance moved. And so you can measure how much this community is changing over time. So how dissimilar is this community becoming? And so then we did this for every single plant across all these inoculation treatment groups. And we found that the temporal variation in parasite community structure differed based off of the inoculation treatments. And specifically, we found that the plants that were infected by Rhizoctonia had communities that changed more over the course of the entire experiment. But then we also found that there was an interaction between Epicloe, which is that fungal endophyte, that mutualist endophyte, and Rhizoctonia inoculation, where you don't see those effects when you have Epicloe infected. And then just to orient you with this graphic, on the x-axis is all the treatment groups, and the y-axis is the total distance moved by the parasite community. So again, this is just how much it's changing over the course of the entire experiment. And so next, I'm going to look at whether the trajectories of these parasite communities differs based off of the inoculation treatment groups. And so when I'm looking at these parasite community trajectories, I'm trying to look at the central tendency of the community. So next, what I did was I took those ordinations that I showed you in the beginning with those messy multi-panel plots, and I calculated the centroid of each community. So basically, this is like the average community. And then we looked at how that changed over time. And so here is our first survey event. So you can see this goes in sequential order. So you can see the trajectory of the centroids of the communities over here, and then it goes all the way to 13. So we calculated the centroid of these communities across all the treatment groups, and this is shown here. And so this just gives you an idea of the central tendency of how these communities are changing. And you can see generally there are differences, and then I'm going to show you the actual analysis and how we approach this. So specifically, we wanted to know whether these communities were diverging or converging towards each other. Because the idea would be if these parasites are potentially interacting, they might have divergent communities or they might converge. And so to compare parasite community trajectories, I'm just going to show you what we did. So if you imagine overlaying these two trajectories, we have over in the purple, the plants that were not infected with any of symbionts, so they have no prior infection. And then in the green, we have the plants that were infected with all 3 symbionts. So both the parasite, the 2 parasites, and the mutualist. And then what we can do is we can see whether these communities are diverging or converging based off of the distance between these community centroids. So here is an example of community convergence. And you can see that the distance between these community centroids are decreasing over time. So that means they're becoming more similar and they're converging towards potentially a similar community state. And then however, over here, we can see community divergence, where the distance between the community centroids are increasing over time. So they're becoming more dissimilar over time. So this means that they're diverging. And so going back to our previous example, we can look at this, where we have, again, our 2 community trajectories overlaid over each other. And then we can look at over time whether the distance between these centroids are increasing. And what we do is we see that there is an increase in the distance between these centroids over time. So this suggests that these 2 communities are diverging from each other. And now just to give you some more examples, we found that the plants that were not infected with any of the 3 symbionts initially tend to diverge from other inoculated plants. So here we're looking at the distance between the negative plants, so the plants that I'm calling naive right now that have no symbionts, and we're comparing them with all the other treatment groups. So over here, you can see, just for an example, we have on the x-axis time and the y-axis is the distance between the centroids, and you can see pretty much across all these treatment groups, they tend to diverge. So you see an increase in the distance between these community centroids, which means that they are becoming more dissimilar over time. However, there is one case where we found no general trend for convergence or divergence. So in the plants that were co-infected with the 2 fungal parasites, Colototricum and Rhizoctonia, these plants did not diverge or converge with the plants that were uninfected. And so basically what's going on here is we have periods of convergence and divergence. And so this could be just stochastic change going on here. And then we can look across all of our other treatment groups. And so basically to orient you over here, if you have a dark blue square, that means we found evidence for divergence. If you have a light blue square, that means there's no general trend because we never found convergence here. And just to point out what I showed you before is that plants with no prior infection exhibit divergent parasite community trajectories. So this is just a summary version of what I showed you before on the other slide. And then another interesting thing was that we found that plants that were inoculated with Rhizoctonia tend to show no general trend for divergence or convergence. So they have that thing where I showed you before where you see convergence, divergence, convergence, divergence, convergence. So there's no general trend. And so we can interpret this as potentially just being more stochastic change. And then again, these plants inoculated with Rhizoctonia show no general trend, even when they're not infected with epicloe. And so we found evidence for divergence between the inoculation treatment groups, but I also need to burst my own bubble and show you within treatment variation in parasite community structure, because we see these general trends for divergence, but there's also a lot of random change and potential drift going on within this system. So we did see within treatment variation in parasite community structure and that it increases over time. So over here on the x-axis is just our sampling time, and on the y-axis is a distance from the centroid. Because in all the other analyses, I use the centroid, and here's just showing that variance from the centroid. So this is just showing dispersion. And so you can think of this as just generally these communities are drifting over time. And so even though we did see a central tendency for divergence, we're also seeing within group divergence. And so in summary, we found that plants inoculated with Rhizoctonia changed more over time, and that we found differences in the parasite community trajectories. But this was only in the case with plants that were infected with no pathogens or epicloe. And that the Rhizoctonia inoculated plants showed no general trend, and we see more so stochastic change. And then within treatments, we found that the parasite communities tend to diverge over time. And so what this got me thinking about is how are we going to balance these stochastic changes of these really colonization dominated systems, and how to parse that apart from these effects of within host interactions and community patterns. And then also thinking about how do we delineate the appropriate spatial and temporal scale to actually detect these interactions in these community patterns. And so with that, I would like to thank the Mitchell Lab, and again, my co-authors, Charles and Brooklyn, and then our honorary lab member, our cardboard cutout of Danny DeVito, and then also our funding sources. And I am done.
Thanks a lot, Rita, for a great talk. We have time for one question. Yeah, there's a question from Mark. Nice talk. The trajectories of the NMDS in many of the treatments seem to travel in a circle, arriving back where they started in ordination space. Any explanation why this pattern exists?
Yeah, so they start off as uninfected, and then everything started to die towards the end of the experiment. So they returned back to uninfected because the plants were dying out, and the epidemics were also starting to decline over time. So that's kind of just naturally what they would do.
Thank you, Rita.
We do have time for one more question if anyone has it, because next up we just have a break until 5:40.
Any other questions?
Does anyone want to know where you can get a Danny DeVito cardboard cutout? Amazon.
All right, well, thank you, Rita, and thank you to all of our speakers in this first half. We're going to take a break until 5:40 and then resume our session then, 5:40 Eastern. So enjoy your break, and we'll see you back soon.
We'll get it going.
So welcome back after the break. I'm glad to see all the people who came back. We're going to start out with, sorry, just let me scroll a little bit, with Nora Schulz, who's going to talk to us about the impact of co-infection on apparent competition.
Thank you. I would like to start with first thanking the organizers for giving me the opportunity to talk, and also for finding the time and energy in these trying times to organize this virtual conference. And before I get started on any content, I also have to mention that part of this presentation, the theoretical modeling, was actually not done by me, but by Faith and Anne. And I hope I can do their great work justice and don't make too many mistakes in presenting their results. So today I will talk about how infection can change the outcome of competition between two species, and what potentially might happen if there is a co-infection with a second parasite. In the Tate lab, we work, our model organism are flower beetles, and they're a major pest of stored grains and can be found everywhere in the southern states, also in India and southern Europe. But this gives us a chance to actually go out in the summer and collect them in these traps. And one of these species we find, Tribodium castaneum, the red flower beetle, is actually a very well-known and well-studied model organism. There's a genome available and CRISPR-Cas and all such things. And then in these traps, we often find the second species, which is called Confusum, because it is so easily confused with the red flower beetle. They can only, one of the very few ways to distinguish them by is like that the antennae are slightly different shaped, and they share these very similar niches. They both have to be resistant against all kinds of pesticides that they're attacked with, and so they're regular co-occur. But already more than 70 years ago, people observed in the lab that they don't co-occur if they're kept under disease-free conditions without any parasites. So this is a study by Park et al. That was reviewed in the late 90s here again. So if they kept together, always castaneum outcompetes Confusum, and Confusum becomes extinct. But when they added Parasite to the picture and infected both species with this protozoan parasite Adelini tribulii, the situation is reversed and castaneum becomes extinct and Confusum dominates the competition. And this is what we call parasite-mediated apparent competition. And in this scenario, so the parasites can have opposing strategies in the way they are transmitted, for example, and so they can facilitate each other or antagonize each other. So, for example, one parasite could already weaken the immune system, giving the second parasite a better chance, or they can compete both for resources within the parasite. So they're very different outcomes of what happens in the co-infection scenario. The parasites we work with in the lab are, on the one hand, protozoan gregarines that reside in the gut of the beetles. And most beetles that we collect in the wild actually are infected with these. They cause relatively asymptomatic infections, so with very low mortality and therefore have a high prevalence. They're just shed through the feces and then taken up by uninfected beetles that become infected. And the second parasite has a very different strategy. So it's an obligate killer, Bacillus thuringiensis, a bacterium which produces endospores and toxins that are able to breach the gut wall of the beetle, and then they have to kill it to shed so that the spores are distributed into the environment and new beetles become infected. So they cause a very high mortality and the prevalence in nature is very low. So we know that to this BT infection, our two beetle species have different resistance. So the Confusum beetles survive aseptic infection with BT much better than the castaneum beetles. This has been shown repeatedly in all our experiments. So we know for the single infection that the infection with BT can change the outcome of the competition between these two species. But what happens if we co-infect with this other parasite, the gregarines? There are three potential outcomes of this. So either it could do nothing, nothing changes at all, or a co-infection could make the difference more severe. So the confusum dominates even more, or it could reverse the competition, and Castaneum could become the winner of the competition again. To investigate this further, we started, or not me, the others in the lab, Anne and former undergrad Faith Rovenold started to work on a model, and the manuscript is currently in review and hopefully published any day now. So to answer the question, how does co-infection reinforce or reverse infection-mediated apparent competition? And this is an SIS model, so a susceptible, infected susceptible model. That means that even after recovery from infection, individuals are not immune but become susceptible again, which is mostly the situation observed with insects. And then there's a generalist parasite, the BT, which is the obligate killer, which infects both species. And then each species has its own specific gregarine parasite. This is just what we observed. So we tried infecting the other species with gregarines collected from one of the beetles, but that doesn't work. So these are specific, but they still share the same life history and cause same disease in both species. And all three parasites need an environmental stage to reinfect new beetles. So we have these three states that were simulated in the model. So there's the disease-free state, where there's the direct competition between the species, and confusum, as I mentioned before, wins in this Castaneum wins in this situation by having a higher reproduction rate. And also another factor that affects direct competition is cannibalism. So the beetles tend to eat their own eggs, but they can't differentiate between the different species, and then they eat more from the other species. And then there is the single infection, where outcome is dependent on the mortality and recovery of each species to disinfection. And then we have the co-infection scenario, where everything can be changed again, depending how the second parasite, what kind of mortality and recovery rate it causes, or how it changes those in response to the focal parasite, the BT. So if you remember that without the parasite, without a BT infection, Castaneum wins this competition. If there is an infection with confusum, because with BT, confusum can now dominate the situation under certain conditions, because it has a higher survival rate of the BT infection. So the resistance is not so important here, so the recovery rate, but more the tolerance. So if confusum becomes more tolerant, there's lower mortality relative to Castaneum. It can outcompete Castaneum. This changes again for very high survival rates, because at that point it's likely that the parasite prevalence is too low to cause any harm to either species, because remember that individuals have to die to shed spores and cause new infections. So what happens if we add the co-infection with the gregarines? If we keep the same assumption that confusum is more likely to survive the BT infection, a co-infection can promote coexistence or inclusion depending on different shifts in host mortality. So what you see here is for host 1, which is confusum, and host 2, which is Castaneum, the relative survival of co-infection compared to the single BT infection. So if you move to the right here, they survive the BT infection better when they're also infected with Gregorines, or they survive it less. And then the same for Castaneum. And we see here, if both survive better under, Confusum survives better under co-infection and Castaneum does worse, then Confusum dominates the picture. And if Castaneum survives better under co-infection than under single infection, and the situation in Confusum isn't changed, then Castaneum can outcompete Confusum again. And then we also have this area where they can coexist. So after this modeling work was done, we also try to see what happens in the lab when we do these experiments. And for this, our lab manager, Sijin, was a huge help and needs to be mentioned here. And so the experimental setup looked at as follows. So we mainly did these experiments only in Confusum. We're still working on the Castaneum part. We had some problems with the Gregorine infections in the Castaneum beetles. So we collect eggs and then we just put them on flour that is mixed with frass that is either contaminated with Gregorine gametocysts or clean flour. And then when we have the larvae, we either we check developmental speed by measuring days they need until pupation, or we take larvae before they pupate and infect them with a lethal dose of BT, and then we have a control for wounding, and then we follow survival for 24 hours. So these bacteria kill the larvae within 12 hours normally. And then we can also, via qPCR, check Gregorine infection status. And also we look later on at BT load, so how severe the infection was. And first, we always check whether the Gregorine infection actually worked. And as you can see here, so these are CT values for a Gregorine primer. So the higher this value, the lower the Gregorine load is. And so at some point it always amplifies. So we set this arbitrary level at 30, at cycle 30 to say everything below is infected and everything above is not, which gives us like 60% infection rate, which across experiments we have seen is higher in the Confusum beetles than in the Castaneum, which might mean that the infection in Confusum with Gregorines is not only more prevalent, but also more severe, which might have some consequences that I'm gonna get to later. Then we saw that the Gregorine infection, even though it doesn't cause any mortality, it still has some costs for the beetles because it slows down their development. So time until pupation, which is shown here, is significantly shorter in the beetles that have not been exposed to Gregorines, and these are the ones that are exposed and mostly infected. And the larval stage of these beetles is just one that is very, unprotected. So for them, it's important to get to pupation as quickly as possible, also to be safe from a BT infection. And so these might be actually some cost of these Gregorine parasites, and this is a possibility to expand the model that we have to include different life stages and developmental time as well. And then we actually have survival data now for both beetle species again, where there were, so the dashed lines are those that have not been exposed to Gregorines, the controls, and then the through lines are those that have been exposed to Gregorines, and we infected both with potentially lethal dose of Bt bacteria. And we see that in both species, the exposed beetles do worse, but while in Castanium, this is not a significant difference. The difference is bigger in Confusum. So the Gregarine exposure is more detrimental to them than to the Castanium beetles, which leads us back to the model originally. So if we look back at this space, again, this is for when with a single infection, Confusum does better than Castanium. This is somewhat reversed when there's a Gregarine infection there, because then the Confusum beetles do worse when they co-infected, while the Castanium beetles on this side, they are doing much more or less the same as before. So we're actually entering here an area where the model predicts that the species should be able to coexist. To summarize this quickly and give you a brief outlook, so we have shown with the model as well as in experimental work that an infection with Bt can switch the outcome of competition between the two species by changing mortality rates, and that the co-infection with Gregarines can actually reverse this effect by leading to a higher mortality from the Bt infection in the less susceptible species, the Confusum, while there's no change in the Castanium beetles. We have some idea this might depend on the severity of the Gregarine infection. Maybe you remember that I said that this appears to be more severe in the Confusum beetles than the Castanium. Currently trying out a few things to test this. We also have some indication that the lower survival of the Bt infection when there is a co-infection might be due to a lower tolerance and no change in resistance. And then next steps that we are planning to do or are currently doing is looking at the gene expression in these beetles during co-infection. We want to run some metabolic assays to see if also these parasites have to compete for resources. And then the model could be expanded in several ways that I mentioned before. And finally, right now, we're not looking at any priority effects, but this could also be done in the model, and more difficultly also in experimental work. But for this, we would have to get the oral infection with the Bt working. And with this, I would like to thank again Anne, Sijin, and Faith for their collaboration on this, and everyone else from the Tate Lab. This work was funded in part by the NSF grant to Tate. And I thank you for your attention and looking forward to your questions. Thank you.
Thank you, Nora. That was a great talk. We may have time for just one quick question if anyone has it.
Well, it looks like everything was perfectly clear, but people can also contact you to ask you questions. So thank you, Nora. We're moving on to our next speaker, which is Anika Wohlleben. And I want to note that she has pre-recorded her talk, but she's here for questions. And so she's going to tell us about the infection of young of year Threespine Stickleback by Cestode parasite.
Explaining to you why Threespine Stickleback are really useful model hosts for us. And one of the reasons is something we call the Stickleback adaptive radiation, where marine ancestral fish independently colonized freshwater bodies after the last glacial maximum. And up in Alaska, where I work, it happened about 12,000 years ago. And what happened is that you can see on the left on this image, we have this marine ancestral stickleback. It has these plates on the side, it has long spines, and these ancestral forms went into the freshwater adapting to new ecological conditions, right? And so we have all of these different forms radiating out. So we have a reduction in plates on the sides, we have a reduction in spine size, sometimes we have different body shapes and we have different eye shapes. So all of these different things evolving and adapting to new conditions. Another reason why these fish are really useful for us is because we can find them in a wide geographic range, basically all over the northern hemisphere. And there's a long history of studying the natural history of these fish. So we know quite a lot about these fish, about their natural history, how they reproduce, and all those kind of things, which is really nice. One thing that I am interested in is a cestode parasite of these fish. It's called Schistocephalus solidus. As I said, it's a cestode and it's tropically transmitted with a 3-host life cycle. The life cycle starts as eggs or the first larval stage, the coracidium, just floating in the water. When this first larval stage gets ingested by a cyclopoid copepod, the second larval stage, the procercoid, develops. And after this infected copepod gets ingested by a Threespine stickleback, the third larval stage, the plerocercoid can develop. And this plerocercoid really grows dramatically inside the Threespine stickleback. Once it reaches a certain size, usually around 50 milligrams, it is ready to reproduce and sexually mature. And then once this whole system, like the infected stickleback, is ingested by a fish-eating bird, the parasites can reproduce in the gut of the bird, either via outcrossing or selfing, and then the eggs are released back into the water alongside with the first species. And this whole life cycle can start from the beginning. One thing that's really interesting about this life cycle, though, is that we can have different fish-eating birds as the last final host, and we can have different copepods as the first intermediate host. However, the second intermediate host needs to be a Threespine stickleback. So we have this very strong host specificity in this position. And another thing that's really interesting about the system is that the parasite Schistocephalus solidus is not viable in the ocean. So it's not viable in environments or basically just in salt water. So that means that when the oceanic stickleback went and independently colonized all of those freshwater lakes, as I mentioned in my first slide, they first encountered this same parasite. And now in these different lakes, they have to find ways of coping with the same parasites. And we can look at how maybe they use different strategies. Because we assume that there is little to no gene flow between these populations in these freshwater lakes, we can treat these separate freshwater populations as statistically independent natural evolutionary experiments, basically. And something else that's really interesting for us and really useful is that current oceanic stickleback populations represent a proxy for the phenotype of ancestral stickleback. So we can also see how these oceanic fish would react to the same threat of the same parasite without ever having it encountered before. And one study that took advantage of that was done by Weber et al. In 2017. And what they did, they took stickleback, Threespine stickleback, and Schistocephalus solidus from locations in Europe and in Canada, and they infected the fish with either foreign parasites or with native parasites. And they also used one oceanic population. So they used Threespine stickleback that are found in the ocean, and they, in a lab experiment, infected those with Schistocephalus parasites. And that is what I want to focus on. This whole study is very interesting, but for today's talk, I just want to focus on that. And what you can see is that we have a very high infection rate. So the proportion of infected fish is between 80 and 90%, which is way higher than what they found when they exposed freshwater fish to the same parasites. So that means that these naive oceanic fish that have never encountered this parasite really have a disadvantage. They don't have the mechanisms they need to suppress parasites. So it means for us really that there is some evolution, coevolution going on where the fish has the selection pressure on finding ways of coping with the parasite. And what's also interesting in combination with this is that we have extreme variation in parasite infection rates in the wild. This is another study by Weber et al. And what they did, they looked at the lakes in Vancouver Island, Canada, and just compared the infection rates. So we have in this little insert, we have the proportion of fish infected with Schistocephalus solidus, ranging from 0 to 80. And we can see for Roberts Lake down here in red, it seems between 2004 and 2016, there were no infections ever found in the wild. However, now if we look at Gosling's Lake, we can see the infection rate in the wild ranges between around 75% to about 20%. So it's fluctuating a lot, it's going up and down. And if you look at this bigger picture, we have about 50 different lakes from Vancouver Island with their infection rates ranging from no infections to 80% infections and everything in between. So we can see that in the wild, even in the freshwater populations, we have really big differences in how infected fish are, how susceptible fish are to this parasite. And that suggests two explanations to us. So either freshwater populations experience different ecological contexts that maybe make it easier for them to suppress the parasite or make it easier for the parasite to invade the stickleback, or the host fish have evolved different levels of resistance to the parasite in different lengths. So looking at immune strategies. And these two explanations are not mutually exclusive. They probably go hand in hand, and it's a bit of both. And this is the main two questions I'm very interested in looking at in my PhD right now, and probably also going forward, because I just think it's fascinating. So to understand this whole dynamic that's going on between the parasites and stickleback a bit better, I decided to look at the infection rate of young of year stickleback. So what that means, stickleback reproduce once a year. Up in Alaska, that's the end of May, beginning of June. The female will lay their eggs, and then shortly after, the babies, the fry will hatch. And I wanted to see if this fry, when they grow, if they get infected before the first winter, which is ice out around October, or if they get infected maybe after their first winter. And I wanted to compare that between lakes we commonly think of as high infection rates and low infection rates. So this is about populations in them. And also how it develops over the winter. I did my sampling in Alaska in the Matsu Valley. So these are the 4 sampling sites I used, just so you can see the proximity to one another. I went out in August 2018. I sampled the juvenile fish, so the young of year stickleback, that was August, so they're probably around 3 to 4 months old. On the same day of collection, I anesthetized them, I killed them, and I just checked for parasites. I went out to the same lakes in 2019 in May. In May 2019, I looked at the 1-year-olds, which should be the same cohort I sampled in 2018 in August, and I looked at their infection rates. I also went out again in 2019 in August to look at the new young of year fish, the new juveniles. And then my plan was to go out again in May 2020 to look at the 1-year-olds from the cohort of the 2019 fish. And then Corona happened, and obviously everything went a bit different than expected. So I have to really give a big thank you to Dr. Jesse Weber, who managed to get me some samples from some of the lakes I sampled in 2019 in August. So I'm really thankful for that, and I'm still working through those samples. But my hypotheses were that already in the young of year fish, I would see differences in the infection rates between high and low infected populations. And so what I found for the cohort 2018 fish, again, I sampled those in August 2018, in Wohlleben Lake, which we typically think of as a high infection lake, we already have about 70% infection rate compared to Cornelius, which we think of as a low infection lake, which has only 20%. So this fits very well to my expectations, to what I was expecting. Now in the cohort 2019 fish, again, I sampled those in August 2019. We have again Wohlleben Lake in red and Cornelius Lake in blue. I also sampled additionally Wolf Lake and Rocky Lake. we can see that still Wohlleben Lake has a higher infection rate than Rocky Lake. However, if you compare the infection rates to the cohort 2018 fish, it is very obvious that in Wohlleben Lake, we dropped from about 70% infection to just around 30% infection. So it just dramatically dropped, which goes along with what I mentioned earlier, that we see this variation between years. And then we have in Wohlleben and Rocky Lake, which are also typically like high infection lakes. We also have higher infections than we see in Cornelius, the low infection lake, but generally also around 30% infection rate. Then my second hypothesis was that the infection rate of these young off-year fish would drop over the winter due to mortality in the infected fish. So the parasite has a really big influence on the whole system of the fish. It takes energy away from all different kinds. So I was expecting that maybe their condition is just worse and they might not survive in the winter so well. In 2018, in the cohort 2018 fish, we actually can see a slight decrease in the infection rate over the winter in the Wohlleben fish. So we can see it dropped from about 70% to about 50%. It's more of a trend than really statistical significant finding, but I think it still fits what I expected. We don't really see any differences in the Cornelius lake fish. But now if we look at the cohort 2019 fish, and again, I only so far looked at the Wohlleben H1 samples. I'm still working through the Cornelius and the Wohlleben Lake samples. Unfortunately, I don't have Rocky Lake samples, but still it's interesting to look at the comparison. But again, so what we see here that in 2019, the infection rate over the winter rose from 30% to about 80% again. So here we actually have the opposite of what I expected. We have an increase in infection rate, and it's really drastic, it's really dramatic. So it will be interesting to see how the other lakes behave. And actually, this finding goes along with a finding by Heinz et al. In 2016. So he looked at the infection prevalence of Threespine stickleback in Cheney Lake, which is another lake up in Alaska in the same area. And he looked at the infection rate of the young off-year fish in August and September 2010, and then again of the 1-year-olds of the same cohort in June. And what he found was an increase in the infection rate over the winter. So fitting very well with what I found in 2019. Now, a few things are to consider here. On the one hand, he has more sampling points than me. I only went out in August. It would have been interesting to also have some samples from September and October. I sample my young off-year fish, immediately kill them and look at the parasites, which makes it easier to detect small parasites than preserving them first in formalin. This is how he worked. And so I might overestimate the infection rate by finding many parasites that didn't really establish in the system yet. And he might underrepresent the infection rate just by not being able to detect all parasites without the movement. I don't know. But generally, I think our findings overlap here, but not here. And I think the main reason basically is that we don't know all the dynamics going on in that system yet. And other work that I'm doing in my PhD right now, I'm looking at the same populations and I'm looking at immune genes and how they are different between infected and uninfected genes. And maybe they can give me a bit more of an idea. I'm also looking at some plankton communities and maybe get a better idea about the ecology. Over that, I want to thank you for your attention, for coming to my talk. If you have any question, please ask me anything, reach out to me. Here is my Twitter handle. This is to my homepage, a link. And I want to thank all of my funding sources, all of my collaborators, especially Susan Frost and John Baker as my PIs, and everyone who made this work possible, a lot of undergrads dissecting fish. And again, Dr. Jesse Weber for being able to provide me some of it. Thank you so much.
Thank you, Anika. Anika is here for questions and we have time for a few of them. So if you have any questions, please post it on Zoom or the Slack channel. We have a question from Daniel Bolnick. Thanks for a nice talk. Do you know how much the fish in the various lakes are eating copepods?
Unfortunately, I don't. I have not looked at the stomach content for some of the populations. The ones I'm dissecting currently, I'm actually looking at the stomach content as well, and we'll get a better idea about that. I have looked at the abundance of cyclopoid copepod compared to other plankton. But yeah, I don't have the data yet on how much the fish actually consume. It's a good question, yeah.
If you have any questions, you can type it up, and I think Anika can, type the response to those. And yeah.
Also, if you didn't understand something because maybe the sound quality, I'm happy to repeat myself.
Yeah. Yeah, I have another question from Lauren Fuse. What is the likelihood that 2019 just saw a stochastic decrease in the frequency of infection due to factors such as reduced bird visitation or another factor? Is it possible that frequency of parasitism is determined more by year-to-year variation than life stage?
Yes, that could possibly be, yes. I think what I was mainly interested in with this is there was only one study so far that I know of that had looked at all of how the infection rate was over before the first winter. Before that, we weren't really sure in those lakes how if they would at all be infected before the first winter. And yes, definitely, it might be more at the time of the year than the yes. And I also have data on the right now that I'm working through the samples from 2020. I will have also data on the 2-year-olds from the 2018 cohort, and I also can compare that to see how that might be changing. Does that help? Does that answer?
We have time for one more question if anyone has a question. Yeah, we have one more from Jonathan. Are there differences in infection rate between the life stages at a given point in time?
For the parasite or for the fish?
I
know it wasn't mentioned in the question, but maybe Jonathan can.
Different fish life stages, sorry.
That's what I'm trying to look at, right? So this is basically together with the study from Heinz et al. That I mentioned, that we're looking at the different stages. I mean, the fish need to be big enough to consume copepods. At a certain life stage, they're too small to consume the first host. So then definitely it is 0, but after that, that's what I'm looking at.
Thank you, Anika.
Thank you.
Thank you, Anika. Next up, we've got James DuBose, who's going to talk about the overabundance of Paraburg-Holderia in the social amoeba D. discoideum microbiome and its impact on the ecological relevance of the farming symbiosis. I butchered a lot of that title, sorry.
Sorry, it's quite a mouthful. Okay, let's see. Okay, can everyone see and hear okay?
Looks good.
Okay, all right, thank you. Oh, hi, my name is James DuBose, and I'm an undergraduate at the University of Central Arkansas, and I'm going to be talking about some of the transmission dynamics of Paraburg-Holderia in social amoeba populations, and how this Paraburg-Holderia symbiont impacts its amoeba host microbiome. So symbiotic interactions are pretty ubiquitous in nature, and as a result, they influence the evolution of many organisms that participate in them. And these interactions are particularly prevalent with bacteria. You know, most things, I think, host bacterial symbionts of some kind. So when discussing symbiosis, it's typically beneficial interactions tend to come to mind, such as the human gut microbiome or the origins of the eukaryotic cell. However, symbiotic interactions actually occur on a spectrum that ranges from parasitism to mutualism. And the duration of the symbiotic interaction, as well as how the symbiont is transmitted, are big driving forces in their trajectory that a symbiont and a host might take together. So one particularly useful model organism for studying symbioses is amoebae. So amoebae feed on bacteria in the soil or in the water or whatever their environment is, which places a selective pressure on bacteria to resist their intracellular digestion. So as a result, some bacteria have evolved the ability to resist this intracellular digestion, and therefore they can occupy inside of the amoeba, thus giving the amoeba a microbiome of its own. So one of the most popular model amoebae are social amoebae, in particular Dictyostelium discoideum. So social amoeba, they undergo a social life cycle that involves a transition from single cellular to multicellular in response to low bacterial food conditions in their environment. So when food bacteria are depleted, D. discoideum secretes a signal that signals thousands of these cells to aggregate together and form a fruiting body that's used in spore dispersal. I was initially thought that these fruiting bodies were cleared of bacteria throughout this social cycle by these sentinel cells that are differentiated amoeba cells that serve kind of like immune-like functions. However, it was recently discovered that bacteria could be cultured from D. discoideum fruiting bodies if the D. discoideum was carrying certain Paraburghalderia endosymbionts. So this phenomenon was dubbed farming because it was reasoned that Paraburghalderia was allowing D. discoideum to carry food bacteria in its fruiting body. So if the fruiting body landed in a soil environment that was scarce in food bacteria, it could then seed the soil with its own bacteria that it carried with. So this was proposed to be a beneficial interaction. However, these conclusions were drawn from laboratory experiments and were somewhat lacking in ecological context. And then it also has to be noted too that, since this discovery of this Paraburghalderia conferred a microbiome, other social amoeba host microbiomes as well outside of Paraburghalderia. So 3 species of Paraburghalderia induce this farming phenotype. There's P. agricularis, P. bonnii, and P. haleyella. And each species differs in what I refer to as symbiotic potential. So this means its cost of infection, it's how much food bacteria it allows the amoeba to carry, the location that it allows the bacterial carriage, and also its prevalence in nature. So our goals of this study were to look at this symbiotic interaction in more of an ecological context. We wanted to study how things like changes in prevalence, how the symbiont is transmitted, how these patterns are established in nature, and what implications that might hold for the nature of the symbiosis. And we further wanted to investigate, is Paraburghalderia actually inducing the carriage of bacteria in nature? Okay, so in order to do this, we would compare Paraburghalderia infected D. discoideum, their microbiomes, to the microbiomes that were uninfected. Okay, so our first question to get at these overarching questions is, how has the prevalence of Paraburghalderia in D. discoideum populations varied over time? So a previous prevalence study had been conducted in the year 2000 in the Virginia Mountain Lake area. So we traveled back to this location and we picked 4 sampling sites. And within those 4 sampling sites, we collected around 10 to 15 soil samples per each of these sites. And then we returned to the lab and we cultured the amoeba from the soil, and we collected over 300 D-Discoideum isolates, as well as some other social amoeba isolates as well. We then used PCR to screen for the Parabricholdaria symbionts, and we used next-gen sequencing in order to get full microbiome composition of around a subset of about 50 infected samples and around 20 uninfected samples. So what we found was that only the prevalence of one, P. agricolaris, of one of these Parabricholdaria symbionts has increased. As we see in the year 2000, it was 14.8% prevalent, whereas in 2019, it was 35.1% prevalent. And then we only found one case of infection of P. bonnii, and then we did not find P. haliella at all. So this could be indicative of some maybe greater relevance of P. agricolaris, or perhaps a higher degree of detriment of P. bonnii and P. haliella. So here's just kind of the distribution of our sample sites. We did find a flat rate of across all locations of about 35%, but these tended to vary depending on site. So we found in some sites as high as 65% prevalence, and other sites as low as 15% prevalence. So it is variable infection rates in nature. So our second question to investigate this kind of ecological framework was, how genetically diverse is Parabricholdaria, in this case P. agricolaris, because that's all we found, in D-Discoideum populations, and how is it transmitted between the hosts? So what we found, based on sequencing, Sanger sequencing of our initial screening sequences, we found that P. agricolaris does exhibit a high degree of genetic diversity. We found 10 P. agricolaris haplotypes, and 7 of which had not been previously identified in the previous prevalence study. Those are the ones indicated in red here. So overall, this paired with the this paired with the prevalence, we're still digging into this ecological framework. So our next question is how is P. agricolaris transmitted? So we found that certain haplotypes infected both D-Discoideum hosts and other social amoeba hosts. So we reasoned that they could not have gotten there from vertical transmission. So P. agricolaris must be horizontally transmitted from one amoeba host to another. So this doesn't necessarily exclude vertical transmission either. In laboratory studies, we do see that P. agricolaris tends to be vertically transmitted as well. It kind of sticks between 5 generations or so. So it is quite prevalent. But overall, our kind of conclusion here is that we see a high degree of genetic diversity, and the environmental acquisition of P. agricolaris could be responsible for this high degree of genetic diversity. So our second set of questions was to search for evidence of this Parabricholdaria-induced farming in nature. So our first question to answer this was, how does Parabricholdaria infection impact the diversity of the D-Discoideum microbiome in nature? So, like I mentioned, we selected approximately 50 P. agricolaris infected samples and approximately 20 uninfected samples. And we sent them for next generation sequencing to get microbiome compositions, and these sequences were analyzed in QIIME 2. So here's kind of our flat results. This is in the red, in this bar across the bottom, we see P. agricolaris infected samples. And then in this gray, we see uninfected samples. And initially, we can see when there's P. agricolaris, there tends to be a lot of it. But we can go to further quantify that with alpha diversity. So we hypothesized that if farming were happening in nature, that if P. agricolaris was indiscriminately signaling these different food bacteria to be carried by its D-Discoideum host, that D-Discoideum infected with P. agricolaris would have a more diverse microbiome than those uninfected. So this here on the right is just a ghost graph of what we'd expect if farming was happening in nature. However, we did not see this pattern across every alpha diversity metric that we looked at. We saw a general same rate. We saw that P. agricularis infected microbiomes were not significantly impacted. Their diversity was not impacted by P. agricularis infection, and phylogenetic diversity was actually decreased with P. agricularis infection. So another way we can look at if this farming is happening in nature is comparing just the flat compositions of the D. discoideum microbiome. So our Parabricoldaria or P. agricularis infected microbiomes more similar to each other than they are to uninfected microbiomes. So we reasoned that, and there's a couple of different ways we can look at this. So we can either consider the relative abundance of the different bacterial members of the microbiome, or we could not consider that. So we hypothesized that if P. agricularis was facilitating the carriage of specific food bacteria, which might be another instance in which farming is occurring in nature, that P. agricularis infected samples would have more similar microbiomes to each other when abundance is not considered. And that's what this ghost graph is depicting on the right, because when abundance is not considered, everything's weighted the same. So we're generally looking at just straight composition, what's there. However, when abundance is considered, it gets a little dicey. We don't necessarily have a flat hypothesis for that because it would depend on the availability of each bacteria in the soil, which can vary by location. So we kind of just left that to see what we see. And what we saw was that when we don't consider relative abundance, which was our primary metric, and this was estimated by unweighted unifrac, we did not see a grouping of these P. agricularis infected microbiomes, which are indicated by the red dots. We saw that kind of this intermixing of all of the microbiomes. However, when we did consider relative abundance, which is estimated by weighted unifrac, we saw a decently strong grouping of P. agricularis infected microbiomes. So we further explored this with another beta diversity metric called robust aitchison, and this is just good at highlighting the important members that contribute towards the clustering of microbiome samples. So we saw that P. agricularis, indicated by this big red arrow on this graph, is the largest contributor bacteria to the microbiomes that it infected. And that's not surprising because when we go look back at that taxonomy bar plot, we saw that P. agricularis was very present. It was very prevalent, consisting of greater than 90% prevalence in a large majority of the microbiota that it infected. So these results were somewhat, unsurprising. So this is kind of a little follow-up question. We kind of concluded, or not necessarily concluded, but our results when comparing the P. agricularis infected microbiomes to uninfected, they weren't indicative that farming was happening. We saw that when P. agricularis was present, that it was very present, probably not leaving much room for a significant amount of bacterial food carriage. However, we phylogenetically inferred the metagenome. So we took the 16S sequences from next-gen sequencing, and we retrieved the whole genome sequences of all the bacteria that were present in our samples. And we looked for gene pathway enrichments or depletions. So we found that P. agricularis samples were significantly enriched in pathways relating to toxin degradation. And this suggests an alternative relevance of P. agricularis, perhaps other than conferring farming. So in conclusion, we saw that P. agricularis is prevalent, yet it is genetically diverse and horizontally transferred. And these traits are not indicative of what we would expect if it was a co-evolved symbiont. So the ability to acquire this symbiont from soil might, or if P. agricularis is acquired from the soil, this would also explain its genetic diversity. Okay, so We also saw that P. agricularis tended to dominate the microbiota that it infected. These results were not consistent with the Farmia hypothesis. However, our analyses did indicate an alternative relevance of P. agricularis to D. discoideum. I would like to thank my lab members, Mackenzie Hoshagan, Eleni Salinger, and Hunter Olson, for bouncing ideas around and helping with sample collection and bioinformatic analyses and things of that nature. And I'd also like to thank my funding sources, the Arkansas InBay program, the UCA Department of Biology, and various undergraduate research stipends that I got along the way. Thank you, and I will now be happy to answer any questions.
Thank you, James. We have time for 2 to 3 questions.
This is not a question, but I just wanted to say that I cannot believe you're an undergrad. That was remarkably well done, so kudos.
I appreciate that. Thank you. I've been working on this project for around 3 years now, so it's I'm glad to see it come into fruition, and I appreciate all my support along the way. So.
We have a question from Joshua. This is a system question, but can you get sterile amoeba, and are they still social if they are sterile?
Excuse me, I'm sorry, my connection is being a little shaky. Could you repeat that?
Sorry, yeah, it's a question from Joshua. The question is, this is a system question, but can you get sterile amoeba, and are they still social if they are sterile?
Yes, so the amoeba are still social if they're sterile. A lot of these experiments demonstrating farming in the laboratory involved antibiotically clearing bacteria from amoeba. So yes, you can get sterile amoeba. Yes. So, and yeah, they are still, they undergo this social cycle in response to cyclic AMP signal. So.
One more question from Andreas. Do you have any idea why the infection rates with different species of Burkholderia vary over time?
Right, yeah, so we're kind of, we see between the different Paraburkholderia species, different cost to infection. So P. bonii and P. haleyella tend to impact the host fitness the most. Well, I guess it's kind of ambiguous. They tend to decrease the host fitness the most in terms of spore production. P. agricularis tends to have the least of this impact. So the prevalence, the changes in prevalence we see could be related to the fact that P. agricularis is less detrimental to its host. We have the genomes of each of these as well. And P. agricularis does have a genome that's over twice the size of the other two. And it still has, biochemical assays have shown that its carbon usage capabilities are comparable to other free-living Burkholderia species. So it's possible that P. agricularis could just be more fit in the soil environment, which makes it a higher probability that it's carried. So yes, I hope that answers your question.
Thanks a lot, James. That was an amazing talk.
Thank you. I appreciate it.
Thank you,
Thanks
James.
everyone. Thank everyone for listening.
Up next, we have Marshall McMunn, who's going to tell us about thermal sensitivity of the gut microbiome in an arboreal desert ant. And then
okay,
Marshall, okay.
See the slides and sound good? Okay, great. Thanks so much to the organizers for allowing me to speak. And thanks also to all my collaborators, including my postdoc advisors on this project, who were Stacy Philpott at UC Santa Cruz, and Rachel Van At at UC Davis. Microbiomes are potentially important in understanding one of the big questions driving ecological and evolutionary research in the 21st century, and that is understanding how these species and ecosystems that we study and care about may respond to these large perturbations. One sort of interesting conceptual model to consider when thinking about how species might acclimate to these changing conditions is considering how microbiome composition could be altered by exposure to a novel environment. And so in this figure, you can see this microbial community on the left being exposed to some novel environment, having altered composition, being the relative abundance of the different bacteria or loss of community members. And then that altered microbial composition potentially altering this shared phenotype of the host and its microbial associates. That alteration of this phenotype could have fitness costs or benefits. And so this microbe-based plasticity could either be beneficial or harmful. And then in some cases where we know that vertical transmission of the microbiome occurs, we might also then go on to look for whether or not offspring share this altered phenotype. And so this could be a potentially really interesting and sort of odd way that species could acclimate to climate change by this sort of passing on of acquired traits through this altered microbiome. So that's sort of the conceptual framework that I was thinking of this project. And the species that we chose to work on is this, has a really fascinating natural history. It is a Sonoran desert arboreal ant, which in and of itself is impressive. Most ants in the Sonoran desert do not live in trees. They live in the ground. And that's for very good reason. It protects them from these extreme temperature variations that occur within days and across seasons. These ants also have an exceptionally poor diet. And so they eat mostly things like plant nectar, pollen, fungal spores, and they opportunistically will take things like mammal urine or bird excrement as sources of nitrogen as well. And they earn the name turtle ants due to their exceptionally thick cuticle, which is costly in nitrogen and also protects them from other ants and other potential predators. They have a talented set of microbes that are associated with several unique aspects of nitrogen metabolism. Some more life history bits about Cephalotes roweri. Queens establish in excavated beetle burrows in tree branches in the desert, including Palo Verde, which are the trees that I use for all of the colonies in this study. The bacteria are very likely vertically transmitted from the queen to daughter ants, and then amongst sister ants within the colony. And one sort of very fascinating bit of natural history, there's actually a filter that develops halfway through the digestive tract that acts as a bacterial filter, sort of separating the midgut and hindgut from potentially contamination from food sources. And so that organ, that bacterial filter, is called the proventriculus. And if you look at this diagram from Lenan et al., you can see this dramatic compositional shift in the bacterial communities as you move from left to right. So the proventriculus is between the crop and the midgut, the blue and the red organs in the diagram above. And as you move from left to right, you can see this dramatic shift into the midgut when it becomes dominated by this taxon in the Opidotales. The genus of that bacteria is Cephalotococcus. So it's has some important characteristics in terms of nitrogen metabolism. There's some more great work in this system that was done by Hu et al., where they compared all sorts of different cephalotes, so a bunch of different members from this genus, but looking at this species in particular, we can see that these bacteria do indeed contribute to the capabilities of this ant in terms of nitrogen metabolism. And so it's able to potentially recycle its own waste nitrogen and the waste nitrogen from birds and mammals using metabolic capabilities that it doesn't possess itself, including the breakdown of urea into ammonia and ammonia into glutamine. So a few questions that we wanted to ask in this system were, how do elevated temperatures alter microbes within a colony? Are bacteria with nitrogen recycling capabilities especially vulnerable? And are alterations of the microbiome permanent, or are they potentially reversible? The methods that we took to address these questions were to go out and locate cephalotes nests in the Sonoran Desert. And so this is near Tucson, Arizona. And we found about 20 colonies in Palo Verde trees. And we wanted to characterize the thermal environment that they were experiencing. And so we, in both summer and winter, stuck a thermocouple inside the nest entrance and measured the temperature variation every minute for several days across these 20 colonies. We did that both in the summer and the winter to characterize the microclimate variation that exists within this particular site in terms of nest temperature. We then characterized the microbiome of workers using 16S amplicon sequencing and qPCR to look at how the bacterial microbiome changes with that microsite. And then we made pairwise comparisons with these repeated samplings from the same nests to look at seasonal change in the bacterial microbiome in the species. We also did a growth chamber experiment where we went and collected ant colonies, put them in artificial nests, and brought them into the lab and put them in growth chambers that differed by 5 degrees Celsius that represented a moderately elevated and a moderately reduced temperature from average. And they were on an artificial diet and exposed to these temperatures for 12 weeks. If we go ahead and look at the results, we can see that in terms of temperature variation in the summer, we see quite a bit of microclimate variation. And the nests do indeed get quite warm. So above 42 C or so inside the nests. And they have strong daily temperature variation. If we compare that to winter temperatures, it's quite a bit cooler in the winter, about 15 degrees Celsius. And just to show you how our growth chamber experiment compares to these temperatures, we were striving to get sort of warm and cool representative nests within the summer. And our 2 treatments fall within natural summer thermal variation. If we go ahead and look at compositional differences between these colonies, we can see that we pick up a weak signal of summer nest temperature affecting bacterial composition within these ants. And so you can see that in this NMDS plot by the sort of moderate color gradient across the plot from dark to light, being from cool to warm. And we also wanted to see if that those shifts in relative abundance across this climatic gradient had a phylogenetic signal. And so whether particular taxa were more or less relatively abundant at particular temperatures. And we did pick up a signal of unifrac distance being explained partially by this nest temperature variable. And so on this phylogeny, we've painted the weighted mean nest temperature that these bacteria were found at. And you can see that some particular clades are more associated with cooler temperatures, here being purple, and some clades are more associated with warmer temperatures, being the yellow or orange. We also found that warm summer nests have less diverse bacteria. So we see a lower alpha diversity in these warmer nests. And then moving on to the seasonal data, we see a few things in this NMDS plot that I'd like to point out. The first is a really stark separation between summer nests, all in the sort of orangish yellow points, and the winter nests in the purple points. So there's strong seasonal shift in microbiome composition. And then notably, there's also much less nest-to-nest variance in the microbiome composition in the winter. And so this could be a sign of dysbiosis in the summer. There's more variable microbiome composition across the population. And then if we look at the relative abundance of a few of the bacterial orders that we see, one notable trend is that the Ophidiotales, which are the most numerically dominant in the mid and hindgut, are reduced in abundance in the summer compared to the winter. And the relative abundance of several other taxa move in the opposite direction. To look at the growth chamber experiment, we, again, just to remind you, took initial colony microbe measurements and post-treatment measurements. And our two treatments were elevated and slightly reduced temperatures. And to look at an NMDS plot, we found two things. One was that bringing the ants into the lab did alter their microbiome. And so we see non-overlap in this plot between the points from the ants that were taken initially and the points that were taken after exposure to warmer or cooler temperatures. And the other thing we see is some modest amount of variation explained by our temperature treatment, in that the orange and black points don't entirely overlap. And so we did see some shifts in composition when we brought these ants into the growth chamber and exposed them to temperatures that were within the range of natural variation. In our growth chamber experiment, we saw a reduced 16S copy number in each ant that was from the warm treatment. And so there were less total bacteria in the warm growth chambers compared to the cool growth chambers. And to try to get at the question of, are these the same bacteria that are changing in relative and absolute abundance? We used qPCR data to scale each particular sample. So then we got qPCR weighted abundance for each amplicon sequence variant, so each bacterial strain. And then we did a regression of that qPCR weighted abundance within these three different data sets that we were looking at. The growth chamber data set, where we had cool and warm growth chambers, the seasonal data set, where we had summer and winter ants, and the microclimate within summer, where we had the natural variation that existed across nests in the summer. And there's a lot going in this plot. So I just want to focus on these two taxa where we actually did see somewhat consistent responses across these temperature gradients. And so in the Ophidiotales, which again is the most numerically dominant of these core bacterial mutualists, we see consistent negative responses with elevated temperature in the growth chamber across seasons. And so they were less abundant in the summer compared to the winter. Also in warm nests in the summer, they were less abundant than in cool nests in the summer. And we saw similar patterns in the Xanthomonidales in growth chamber and season, but we did not see those patterns replicated over the summer. if we ask the question, if it's the same particular bacteria, the same strains that are responding more or less severely in these different situations, we could take the slopes from that previous plot and compare them. And so each dot on this plot is a bacterial strain, and we see the change in read abundance per degree Celsius colored by bacterial order. And in this plot, if we see points in the same quadrant, in this lower left quadrant, then they're both responding negatively, or the strain is responding negatively in both situations. And if we see them along a line, then they have a correlated response. So we're seeing with this most extreme point in the lower left, that bacteria was most heat sensitive in both the growth chamber, and then also it was the bacteria that was reduced the most in abundance in the summer in the field. So both the Ophidiotales and the Xanthomonodales experienced this sort of correlated heat response, which to us suggests that some of the seasonal change is actually due to this reduction in abundance due to temperature. And just for the purposes of time, I'll skip this, but microclimate was similar for Ophidiotales, but not Xanthomonodales. And to summarize, we see a strong seasonal change. There's reduced abundance of some of these common core mutualists. They're likely reversible. So we saw a recovery in the abundance of these Ophidiotales in the winter. And the similarity of nests is greater in the winter. All of these things adding up to say that summer is probably bad for this microbiome. We see some microclimate effects, but they're relatively weak. And these are pretty tightly correlated across ASVs in the response in the two settings. We weren't able to determine if these shifts in microbial compositions affected phenotype, but we would have to guess that it was negative in the summer, a negative effect on fitness. And we think that these are probably reversible changes. And so these may not actually be inherited since they seem to be reversible changes since we measured summer and then winter, and we saw this sort of more robust, more similar community after it was this sort of dysbiotic variable community. And with that, I'd like to thank all my collaborators and in particular the National Science Foundation for funding.
Thank you so much, Marshall. We have time for one or two questions. We have, we have one question from Joshua. Are there differences between the microbiomes of different ant castes?
Oh, that's a good question. Yes, we sequenced majors and minors as a subset of this project. I haven't looked at that data much, but I remember the Pseudomonodales being the major different bacterial taxa. So there are compositional differences, but we haven't looked in great detail at that data.
The second question is from Scott Powell. What kind of upper thermal limits do you think might exist in this host microbiome microbe interaction? Are there current habitat or thermal extremes that might cause microbiome collapse?
That's a really good question. It seems like they are sort of right at the edge of what they can tolerate in some of the nests in the summer. And so I would say perhaps their range of microclimates they could occupy could be reduced as temperatures increase, or they might have to shift their broader geographic range to then be able to occupy a large set of microclimates again. But it doesn't seem like, as I suspected initially, that microbiomes would be a good sort of adaptive mechanism. It seems like perhaps more of a point of susceptibility for the species.
Thanks a lot, Marshall.
Thank you.
Thank you, Marshall. Okay, up next, we've got David Paez. And this will be another recording. However, he is here to answer questions. And so he will tell us about implications of specialization of a salmonid rhabdovirus for disease management.
So my name is Dr. David Paez, and today I'm going to be talking about specialization of a salmonic rhabdovirus and its implications for disease management. This is a collaboration with multiple PIs from these institutions. So hatcheries and aquaculture facilities usually have conditions that are favorable for disease spread. This is a typical hatchery picture where each one of these race weddings have tens of thousands of individuals. That high density can favor the spread of diseases. One disease that can spread in these hatcheries of North America is infectious hematopoietic necrosis virus, which causes high mortality in salmon and trout hatcheries. Anywhere between 30 and 80% of fish may die once an epidemic of HMV starts. This pathogen is diverse with genotypes that cluster into genetically similar genogroups. This is the phylogenetic tree of IHN in North America, where we have three main groups, L, M, and U. And these groups are further resolved into genogroups that are commonly observed. So within the M group, we have the MD genogroup, which is a collection of genotypes that are commonly observed in our study system. And within the U group, you have the UP and the UC genogroups. Field observations and other laboratory data suggest that the genogroups are associated with some hosts, and that suggests that there's different levels of specialization and journalism. L is associated preferentially with Chinook salmon in Northern California and Southern Oregon. MD is associated with steelhead and rainbow trout in the Columbia River Basin. UP is often found associated with sockeye salmon throughout the northern distribution of sockeye. And UC is interesting because it mostly occurs in the Columbia River Basin, but it's often found associated in all three species. And so these led to the hypothesis that the different genogroups L, MD, and UP are host specialists, whereas UC is a host generalist. And so the aim of our grant is to test whether the field observations support theory of the evolution of pathogen specialization and generalism. And we address this objective using multiple approaches. We look at virus fitness measures, and host responses using laboratory experiments. And we combine these experimental data with field data and models to produce long-term infection dynamics. And so we basically evaluate the infection process at every stage, from the entry to early infection, post-survival, and the long-term effects of clearance and persistence. And today I'll be using results to test whether the virus fitness of early replication and long-term persistence support this idea of specialization in generalism. And then I'll be using these results to test whether specialization and generalism can affect the transmission dynamics and fish mortality observed in hatcheries. So to quantify early virus replication, we conducted an experiment over 14 days, a factorial experiment where the three hosts were exposed to the four genogroups with 3 strains per genogroup. All fish are exposed at day 0, and at each one of these sampling times shown by the red arrow, we sampled 5 fish to measure the viral load. In the long-term persistence experiment, it ran up to 8 months. And again, it was a factorial experiment where 3 hosts were exposed to 3 genogroups with only 1 representative strain per genogroup. All the fish were again exposed at day 0, and an increasing number of samples of fish were sampled at these different times. And from that data, we measure the positivity rate and the viral load, although I won't be talking about the viral load for this. And so from these early virus replication experiments, we hypothesized that the specialists should replicate best in the host to which they best adapted to. And so we would expect to see high peak viral loads of the hypothetical specialists in their adapted hosts. And in this figure, we have the genogroup on the x-axis and the peak viral load on the y-axis, and each panel being a different host species. The colors represent the different strains that belong to the genogroup, and so they're all clustered according to the genogroup. And we observed indeed that the hypothetical specialists do have the highest peak viral load in each one of the hosts. In steelhead trout, the MD, which is a hypothetical specialist, has a very similar viral load to UC, even though it's still a little bit higher. It's comparable to the peak viral load observed for UC. In terms of the long-term persistence, again, we hypothesize that a specialist should exploit the host to which they're best adapted to for longer. And indeed, we observed that L persists for longest in Chinook salmon. MD similarly has the longest persistence in steelhead trout. And in sockeye, both UP and UC have a similar persistence profile. When we now look at the data for the generalist, in this case, we're actually hypothesizing that the generalist should have a similar replication across the host that it infects, so a high peak viral load across all the hosts. And so when we look at the data, we see that next to the hypothetical specialist, the generalist does have the second best peak viral load in each one of the hosts. And when you compare the peak viral load across all hosts, this genogroup you see, the hypothetical generalist has the best or the highest peak viral load. So this supports the hypothesis, again, that UC is a generalist. When we look at the long-term persistence profile, UC doesn't do as well as the hypothetical specialist in Chinook salmon or in steelhead trout. But in sockeye salmon, the persistence profile of the specialist and of the generalist are very similar, the persistent profiles. And so how do we now use these data to understand diseases in hatcheries? So we would like to test whether these data on specialization and generalism affects the transmission dynamics and the fish mortality that we observe. And so to do that, we constructed a model where we assume that the infection process starts at time 0, and infected individuals infect healthy, susceptible fish at a rate that's proportional to the transmission rate. Here described by the beta symbol, the beta letter. Infected fish can then either recover at a rate equal to gamma, or they can die at a rate equal to mu. So this model is basically tracking the number of susceptible, infected, recovered, and dead individuals in time. However, to get clean or precise estimates of these transmission parameters, of the different parameter values, we usually need data that on the number of infected, susceptible, recovered, and dead individuals. However, in hatcheries, we often don't have access to all of this data. What usually happens is that people go out and they conduct mortality counts, and that's the data that we have. So we need to infer these infection parameters based on a limited data set. And so to improve the estimation of these parameter values, what I did was to inform these recovery and death rates using the experimental data that I just presented. We use the experimental data of persistence to inform the recovery process, and some additional mortality data to inform this mortality process. And by informing these two processes here, the hope is to improve the estimation of the transmission. So we have data for 2 hatcheries, shown here with the colored symbols. In the first hatchery, we have the cumulative mortality over time for an epidemic of steelhead with MD, which is a specialist. And in this second hatchery, we have an epidemic of Chinook salmon with UC, which is the generalist. And so even though, and you would notice that in this second hatchery, the epidemic is kept short of 12 weeks. And that was due to a management decision where they decided to cull the fish, given that the epidemic was getting out of hand. However, this data is very valuable because it's not, although it's very common to observe epidemics of MD in steelhead, it's not very common to see epidemics with UC. The infection process has a much lower disease expression, and therefore it's not commonly observed in the field. So these data are very valuable. One of the objectives of the model is to reconstruct the rest of this epidemic trajectory in order to test for differences in the epidemic duration between these two hatcheries. And so the gray lines here show the model fit to the data. And it basically suggests that the model is doing an adequate job to explain the data that we observe. When we look at the estimates of the transmission rate, we find a trend where the transmission rate of MD in steelhead is slightly larger than the transmission rate of UC in Chinook salmon. However, the credible intervals around these estimates overlap each other, so it's hard to draw any conclusions with a statistical certainty. And these high credible intervals are certainly due to the lack of data in the key processes of infection and recovery. However, we can then take these average estimates of the transmission rate to simulate a model in order to reconstruct the entire epidemic trajectory and test whether these two hatcheries differ in their epidemic duration. And so in this figure, on the x-axis, we have the epidemic duration, which is the number of weeks until no additional infected individual is observed. And in the y-axis, we have the proportion of simulations. And so a value of 0.1 that intersects a value of 10 basically suggests that 10% of the simulations have an average epidemic duration of 10 weeks. And so we can see that there are differences in the epidemic duration between the specialist and the generalist, suggesting that it takes the epidemic lasts for a little bit longer for the specialist compared to the generalist. So this is an important insight that we're getting from a model that is informed by experimental data and the hatchery data, which we really couldn't gain access to if we didn't have the combination of these different data sets. So in conclusions, we have that pathogen fitness differences reveal the outcomes of the evolution of specialists and generalists. We have that life history variation of pathogens can lead to different disease outcomes. And this is interesting because this result is suggesting that evolution, the evolution of the different infection strategies of pathogens can affect how we manage diseases out in the field. We are currently using additional landscape data to increase our certainty of the transmission process. And with that, I would like to thank you.
Thanks a lot, David. We have quite a lot of time for questions. So please type in if you have any questions.
Sorry, David, I think you're suffering the effects of Zoom fatigue over the course of 2 days of conference. It was an excellent talk, though. I don't have any great question to ask, but I enjoyed it.
That's cool. Yes, I think everybody's a little bit zoomed out.
Yeah, I mean, it happens.
Mm-hmm.
I can ask a question that's not very insightful about the science, but I've always worked with species that are not of conservation concern or of general interest to people who might want to use them for food or for fisheries. And so what is that like, working with species that are important economically? Does that put a lot more pressure on you as a researcher to make sure that your data is really tight before you share it? Or is there anything that you have to think about with that?
It certainly puts a lot of pressure in that sense, but I mostly see it as a great opportunity to kind of showcase the evolution of pathogens and life, the importance of life history evolution for understanding disease ecology. So it certainly, I mean, I think everybody should be equally as critical about their own data, regardless of whether the species is important or not. But it certainly does make you double check more than once that results are solid and that everything is tight. But I think I see it more as an opportunity to showcase, given the amount of interest. That it's an important system, and with salmonids, they're ideal to study these disease processes because of like their conservation efforts have basically created this system in Pacific salmonids to rear fish in massive densities in like really small spaces, and so that brings disease from outside, and it creates all sorts of additional problems. So. It's kind of an ideal system to showcase all of these important questions in the interface between ecology and evolution.
And I guess that the life history knowledge is off the charts too. If it's a species of economic interest, then that life history work is probably already done in a lot of ways. That's cool.
Right. Yeah. And like, for example, for this pathogen, we are trying to understand its life history from when it enters the host till a couple of months or even like 8 months later when it actually leaves the host. And we can do that because we get money to study this really important conservation issue in salmonids. And so, yeah.
Well, thank you so much. So our next speaker is Daniela Pinto-Coelho, who this is also a recorded talk, but she's here to answer questions when she's done. And the talk is on network analyses reveal the role of large snakes in connecting feeding guilds in a species-rich Amazonian snake community.
Interaction between species are a key component for understanding biodiversity. In fact, individuals of all species rely upon ecological interaction to obtain food, to breed, or to protect against natural enemies. Ecological interactions form networks that connect populations of different species in a locality. The organization of these networks may have important conservation conservation implications, potentially affecting the robustness of ecological system to species loss. In this context, it is essential to understand how factors that influence the interaction between individuals affect the structure of networks at the level of ecological communities. Among the traits that may affect network structure is body size, which is directly associated with the ability of individuals to consume resources. If the larger the predator individual, the greater its ability to kill larger prey. We should expect, at the species level, larger average body size to be correlated with a larger number of resource type consumed by predator species. Furthermore, if only body size were influence the capacity to consume a wider range of resources, it is expected that the diet of the smaller predator species will be a subset of the eatings of the larger predator species diets, leading to nested ecological networks. On the other hand, food resources are not homogeneous distribution in environment, and the degree of specialization in the consumption of distinct sets of prey may require distinct adaptations. Thus, we can expect that due to the restriction related to prey handling, prey detection, or nutritional yield, predators of bigger size are predisposed to discard smaller prey, generating deviation from the nestedness pattern, and possible forming semi-isolated groups in the network, leading to modular ecological networks. Here, we explore the trophic network organization of a rich and well-studied community Amazonian snakes. Many snakes are top-level carnivores and perform an important role in community ecology. We characterize the structure of the interaction networks between snakes and their food resources. We expected that if only snake body size were shaping networks patterns at the species level, the structure would be nestedness. On the other hand, if specialization in resource consumption were driving patterns of resource use across snakes, modularity would be expected. We then evaluated the role of different snake species and the effect of snakes' habitat use, referred to here as a lifestyle, in shaping the network structure. We analyzed the snake's diet derived from a long-term study carried out in a central Amazonian site on the natural history of forest snake. We described the resource used by snakes as a binary interaction matrix, and our food resources are not described at species level, but at coarse categories such as lizards, frogs, small mammals, birds, and mollusks. We opted for these coarse categories because the evidence that snakes are specialized in broad categories of resource, for example, serpentiform organisms that include snakes, amphibians, and caecilians, and because the level of detail available from the diet analysis of snakes. We used 4 metrics to characterize the structure of the interaction networks. Degree distribution, which is the description on how the number of food resources a given snakes can feed on varies across snake species. Connectance, which is the proportional of all possible interaction actually recorded in the network. Connectance values range from 0, non-connected networks, to 1, maximum connectance. Nestedness, which consists of an interaction patterns in which the specialist interaction with sets of resources with which the generalist also interact. Nestedness values range from 0, non-nested networks, to 100, perfect nestedness. And finally, modularity, which is a measure of the extent to which the network is formed by groups, in which snakes within a model overlap in much of the resource, whereas snakes in different models show no or weak resource use overlap. Modularity values ranging from 0 non-modular networks to 1 completely nodular. If the network of interaction analyzed follow the organization pattern structured by body mass, we hypothesized snakes' average body mass to be positively correlated with the number of resources consumed by the snake's species. To explore this prediction, we investigate the association between average body mass and the role of species in the network structure. In order to understand the individual contribution of each species of snakes to nastiness, we use a jackknife resampling approach in which we remove a snake's species and recompute the degree of nastiness. We repeated the procedure for all snake species in the networks, and then we computed the change in nastiness, where the delta NE is the degree of nastiness of the complex network subtracted from the degree of nastiness after the removal of snake species E. If body size is shaping the contribution to nastiness, we should expect that the delta NE will assume increasingly positive values as larger snakes are removed from the networks, indicating that nastiness is higher in the presence of those largest snake species. In results, we recorded 163 interactions between 62 snake species and 26 food resources that were heterogeneously distributed among snake species, where many species have few interaction and few species are very connected. This is the graph of the networks, where the circle represents the different species of snakes, the triangles represent the food resource, some photos illustrating the types of prey, and the lines represent the interaction. The color represents the food models. We can see that lizards and frogs were the most consumed food resource by snakes. And the earthworms, insects, and mollusks are consumed by snakes that are in isolated or semi-isolated models. The network structure shows moderate connectance, indicating that, given the variety of food items consumed by snakes, represented by brown triangles, the snakes analyzed use on average 10% of the resource, a subset of the resource. These results suggest that most food resources may not be accessible to most species, suggesting forbidden interaction associated with possible restriction related to lifestyle and as well as body size. For example, arboreal snakes have morphological adaptations, such as slender body and long tail, which may represent limitation to the consumption of large prey, such as mammals. The snake's resource networks also show significant nastiness, indicating that 33% of the interaction of the less connected snakes species represents a subset of the interaction of the most connected species. Our results supported the prediction that there is a positive association between the number of resources consumed and average body size, indicating that in general the larger species of snakes showed a greater number of food interaction. We highlight that among the larger snakes in the networks are the larger constrictors of the boid family, represented by red circles, that consume a great variety of food resources, such as the largest species in the networks, the anaconda. The correlation between degree and average body mass suggests nastiness was driven by body size. In fact, there is a positive correlation between delta nastiness and average body mass, as well as between delta nastiness and degree, indicating that the larger snakes have a greater contribution to nastiness. One of the explanations of the nastiness patterns found in our study was the larger size variation among species present in the networks. The variation in snakes' body size has led to a trophic hierarchy in which larger predators prey upon more resource items than smaller predators. This hierarchy was detected in several predator-prey interaction networks found in nature. This pattern indicates that predators have the potential to add resources sequentially as they increase in size, although this increment of larger resources may lead into the rejection of smaller, less nutrition, or difficult to handle resources. Finally, the networks also shows significant modularity, with the presence of 6 food models, indicating that the number of interactions with each model is 51% larger than what is expected for networks with the same number of models, the same number of interactions per species, but with random interaction between species. Because dietary specialization in snakes can be related to habitat occupation, we expected snakes' lifestyles to affect the degree of dietary specialization. For example, an aquatic snake would rely upon aquatic prey. If this is true, the distribution of lifestyle in the different models will not be random. We evaluated this prediction using 2 analyses. First, we analyzed the frequency of snakes' lifestyles in different models. We estimated that the possibility of the observed number of species of a given lifestyles in a given model be reproduced by randomly assigning species across models. Then, we analyzed the dissimilarity of lifestyles between models. Similarity between a pair of models range from 0, models are identical in the composition of lifestyles, to 1, no lifestyles occur in both models. In fact, models were associated with particular lifestyles, as indicated by significant or marginally significant probability values present in most models. The most similar models were models 1 and 3, that have the largest variety of lifestyles and food resources consumed by snakes. The model 3 was composed of generalist species in diet and lifestyles, such as snakes of the Wohlleben family. Moreover, the formation of groups based on diet and lifestyle occurred by species that specialize in the consumption of certain food resources, probably associated with their lifestyles. For example, the smallest models, the number 5, was formed by only 2 arboreal species of the genus Gypsas that feed exclusively on mollusks. The model 0s, the most dissimilar model, grouped fossorial snakes, such as the species of the genus Atractus, that are specialists in prey upon earthworms. The modular structure in ecological networks may be associated to factors such as the degree of specialization among interacting species, habitat heterogeneity, the phylogenetic relationship between species, the convergence in the set of species traits, or by a combination of factors. We found that the consumption of specific resources is associated with more peculiar lifestyles. For instance, morphological adaptations to fossorial habits hinder the consumption of prey larger than the snake's head size. Accordingly, arboreal habits impose physical limitation on snakes' morphology and may restrict the consumption of larger prey, such as small mammals, favoring a diet based on lizards and or frogs. Thus, which suggests that the modularity of the networks has emerged from the relationship between the lifestyles of snakes and the consumption of resources restricted to the habits used by the species. To finish, our research revealed an intricate networks patterns resulting from larger snakes feeding on higher diversity of prey and therefore promoting nestiness. Whereas snakes with specific lifestyles and feeding on distinctive reserves promoting modularity. Species removal simulation indicated that the nesting structure is favored mainly by the presence of species of the Wohlleben family, which because of their large bird size and generalist lifestyles connect models in the networks. Our study highlights the particular ways traits affect the structure of interaction among predator and prey at the community level. I am grateful to the CAPS Research Development Agency, to the postgraduate program in ecology at University of São Paulo, to my lab, to my advisor, Paulo Guimarães, and to the collaboration of this work, Márcio Martins and Laura Alencar, and to everyone who is watching this talk. Thank you.
Thank you, Daniela. That was a great talk. We have time for 1 or 2 questions. We already have questions coming in. The first one from Freya. Excellent talk with interesting results. In the regression models, it looked like the anaconda points might be outliers and perhaps driving the relationships as by far the largest snakes. Have you tried removing anacondas and seeing if your results are the same?
Yeah, I tried removing anaconda and the largest, the 7 largest species of snakes, and observed the results, especially about the nastiness. And when I removed only anaconda, the result didn't change. But when I removed the Wohlleben snakes, all Wohlleben snakes, the result reduced significantly.
And second, and the second question by Joyce. Nice talk. I was wondering if besides body size, the constriction feeding behavior could be a key factor for higher diversity of prey, or are these factors related?
So there is other snakes that have constricting habits, but the Wohlleben snakes is a real constrictor snakes, and because of this, I agree that the feeding behavior can be a key factor for higher diversity of prey, because favoring the bigger size and effective capacity of killer larger different types of prey. Yeah, I agree.
Well, thank you so much.
Thank you so much.
Okay, before I introduce our last speaker, I just want to remind everyone we have 3 virtual talks that are available. There are links in the program, so don't forget about those. But without further ado, let me introduce our caboose of the session, John Manube, who's going to talk about specialization within a feeding guild, differentiated diet between co-occurring army ants as explained by contrasting sensory responsiveness to prey.
Thank you, Freya. How's my audio?
Your audio is great, but
Excellent.
I cannot see your screen if you're trying to share.
Hello, I'd like to start this talk today by thanking ASN for giving me this opportunity to speak with you all today. My name is Abe Manubai, a PhD from the George Washington University. And today I'm going to be talking to you a little bit about some of the dissertation research I completed under Dr. Scott Powell, also of the George Washington University. biodiversity. Specifically, we'll be talking about differentiated diet between co-occurring armiids being explained by contrasting sensory responsiveness to their respective prey. To set the stage for you all a little bit, the field component of the dissertation research presented today was all conducted on Barrow, Colorado, an island in Gatun Lake, Panama, formed from the flooding of the canal zone over 100 years ago now. Now, the island is relatively small, only about 15 square kilometers, and to put that into perspective, that's roughly 4 times the area of Central Park, New York City. The island itself is covered in seasonally dry tropical rainforest, and on my very first visit to Barrow, Colorado Island early in my dissertation, I was struck by the many thousands of species that successfully have coexisted on Barrow, Colorado since the flooding. And as an entomologist by training, I was equally struck by the abundance and diversity of different armiid species on Barrow, Colorado Island that have been living there ever since the flooding of the canal zone. As a bit of a primer on armiid biology, because they are quite different from other ants, all armiids are obligately nomadic and obligately group foraging, with raids of some species being up to 15 meters across at the raid front, with a trail of continuous bidirectional traffic back to the nest site, which may be 100 meters or more behind that raid front. I want to note here that all army ants are largely blind. And I want to emphasize this specifically because as human beings, we are very visually oriented. Ants, and especially army ants, are largely chemically oriented. Additionally, all females in all army ants are flightless throughout their life, with morphological adaptations for running, a somewhat unusual condition amongst the ants. And yes, for those of you who were thinking back to our dispersal talks here at ASN yesterday, that means that the females of this species do not leave Barrow, Colorado Island throughout their life. They do not disperse across the water. However, the males do fly and do disperse. I also want to point out that army ants are ecologically important predators because previous quantitative studies of army ant diets on Barrow, Colorado Island by Dr. Scott Powell show that some species have a population level prey intake rate on this island of 2 kilograms dry weight of other arthropods every single day, with similar prey intake rates estimated for other species that have not been quantitatively measured.
Given this enormous rate of arthropod consumption, I questioned how was it possible that at least 20 army ant species are successfully coexisting on Barrow, Colorado Island since the flooding of the canal zone. Interestingly, most tropical lowland forests where assemblages have been surveyed have found a similar number, roughly 20 army ant species coexisting successfully. Now, one potential way coexistence can be facilitated is through resource partitioning, particularly of prey resources when we're talking about these types of predator feeding guilds. And it's important to understand the mechanisms of resource partitioning, especially in these otherwise ecologically similar feeding guilds. Please note that in the composite image here, 2 army ant species in the top 2 panels are carrying their preferred prey items, that is the brood or the young of other ant genera. The focal taxon for my dissertation research are army ants in the genus Aceton, which are largely specialist predators of other ants. They tend to be so specialized they usually only eat a handful of other ant genera. In the top row of images in these slides, you can see common Aceton species which co-occur on Barrow, Colorado Island, and below them you can see some of their preferred prey ants and the approximate amount of their diet that they make up. There is one major exception to this trend of specialization in the genus Aceton, and that is the army ant Aceton burchellii. Aceton burchellii has the most generalized diet of any army ant in the genus Aceton, with about 50% of its total diet being seemingly random litter arthropods,
things like katydids, cockroaches, tarantulas, and scorpions. However, the other 50% of its diet is almost entirely composed of one genus of ant, the Camponotus ants. Now, clearly, these organisms are partitioning their prey resources, but the sensorial and behavioral aspects governing this prey choice remains poorly investigated. Army ants may be using a number of non-mutually exclusive mechanisms to locate these specific prey ants in the complex tropical forest environment they inhabit, but few had been tested to date. While investigating these sensorial and behavioral mechanisms of prey partitioning early on, we found that Eceton hamatum use odors to identify their known specific prey. We found that these odors triggered both increased localized recruitment and slowed advancement of the raid as the Eceton army ants targeted those odor sources. Additionally, we found behavioral responses by the Eceton hamatum to be negligible or zero in response to non-prey odors. This showed that Eceton hamatum, at least, are not interested in the odors of any old ant that they encounter in the environment, but are able to identify their specific prey by its smell. Additionally, we found that not all types of prey odors are of equal importance for the army ant Eceton hamatum to identify its prey and direct its foraging. Using these behavioral metrics as proxies for army ant interest, the army ant recruitment rate and army ant running speeds, we found that Eceton hamatum had the strongest responses to nesting material of their confirmed ant prey, with progressively weaker responses across other odor sources, for example, live ant odors, dead ant odors, and the odors of volatile alarm pheromones from their prey, respectively. In other words, hamatum is most interested in odors indicating proximity to the prey ant brood located in the nest, and secondarily to stimuli rich in prey cuticular hydrocarbons, which are less volatile odors that are associated with the cuticles of many arthropods. I wish I had more time to talk about how we figured this out, so please check out our Journal of Animal Ecology paper from 2020 if you want to know more about this research, or chat with me after the talk. This background brings me to what I wanted to talk to you all about today, a comparison of prey detection mechanisms in co-occurring species of Eceton. So we know now that resource-based partitioning contributes to production and maintenance of biodiversity by reducing competition and allowing otherwise ecologically similar organisms to coexist. We know that army ants in the Neotropics are specialist predators of arthropods, largely of other ants, that partition their prey niches by specializing on different ant genera. In this study, we hypothesized that reduced niche overlap between two co-occurring army ants in the genus Eceton, in this case Eceton hamatum and Eceton burchellii, may be achieved via their contrasting sensitivities to several stimulus modalities, resulting in differing behavioral responses while foraging for potential prey. We addressed two predictions in these co-occurring army ant species, the first being Eceton burchellii and Eceton hamatum share a similarly specialized olfaction mechanism, allowing for detection and identification of their respective ant prey, and two, that Eceton burchellii, with its greater diversity of prey types, may show a greater sensitivity to vibrational stimuli than Eceton hamatum, allowing it to detect and attack non-ant prey. So allow me to paint a simple picture of our experimental design. We would go out into the tropical forest on Barra, Colorado Island every day. I find and identify army ant raid columns as they were foraging throughout the undergrowth. We would then present the army ant raid front with sensory stimuli from different non-prey and prey ants. Some of those sensory stimuli would have a moving organism, some would have motionless organisms. And then we would film the army ants to determine interest in and responsiveness to these different types of sensory stimuli from both known prey and known non-prey. Behavioral metrics like recruitment rates and forager running speeds were used as proxies for interest in these stimuli, with higher recruitment and slower individual running speeds indicating a high level of army ant interest in those stimuli sources. To be a little bit more specific about these stimuli sources, a moving ant stimulus was 1 gram of living ants with their limbs cut to prevent their retreat. In nature, many ants retreat in the presence of army ant raids. This provided the army ants with a combined movement and odor stimulus, movement in the form of the ant struggles, odor in the form of whatever volatiles are being actively released, and additionally any non-volatile compounds associated with the cuticle of those ants. Motionless ant stimuli was similarly 1 gram of ants that had been very recently killed via flash freezing and then bringing them back down to ambient temperature. Now, the motionless ant stimuli provided ants with a very simple odor cue stimulus, simply the odors associated with the cuticle, the cuticular hydrocarbons of those ants, without any movement cues and without any actively released volatiles. Additionally, each army ant was presented with both moving and motionless non-prey sources, ants that no army ant is known to eat, and moving and motionless sources from known prey items, which are different for Eceton and Hamatum, as you can see here. Now here's what we found. Eceton burchellii recruits far more than Eceton hamatum to moving non-prey ants. These are ants that we know that burchellii and hamatum do not eat, but burchellii for some reason recruits more to them when they're moving than when they're not moving versus Eceton hamatum. Both of these army ant species assessed here have very robust recruitment to their respective prey, regardless of whether the prey was moving or motionless, suggesting they have a shared mechanism of chemical sensing of these ant prey types. You'll also notice that the stimuli of prey are elicited from prey are always much greater in magnitude. In this case, recruitment is much greater in magnitude for both species in response to prey items versus non-prey items. We see a similar pattern when we're looking at mean army ant running speeds, in which Eceton burchellii individuals slow down far more than Eceton hamatum individuals do in response to stimuli from moving non-prey ants. Again, this is an ant that neither burchellii nor hamatum are known to eat, but burchellii is more interested relative to hamatum than for the moving non-prey. Also note that the mean individual running speeds are largely similar between army ant species in response to motionless non-prey and moving prey, and both army ant species show considerably more interest in the presence of prey ants, regardless of whether the prey is moving or motionless. The results on the previous slides had me thinking, if olfaction is the only sense that matters for prey detection in Eceton burchellii, then that might mean Eceton burchellii can react to a multitude of different odors from an incredibly diverse set of prey. Recall, as I mentioned earlier, that Eceton burchellii is special amongst the army ants in that it has an incredibly generalized diet. About 50% of the total diet of Eceton burchellii is seemingly random litter arthropods, things like katydids pictured here, cockroaches, tarantulas, spiders, etc. Given that we saw Eceton burchellii is more interested in movement of known non-prey ants than its congener Eceton hamatum is, perhaps Eceton burchellii is actually more sensitive to mechanical stimuli or vibrational stimuli made by the struggling non-ant prey that the swarm encounters. In order to get at this question about exactly what sensory stimuli burchellii is using to identify and attack non-ant arthropods, we conducted a similarly designed experiment in which I would go out into the forest and present Eceton burchellii swarms with 2 types of stimulus modalities. One from a moving, living katydid, and one from a motionless, dead katydid. The moving katydid presented the army ants with a combined sensory input of odor from the cuticular hydrocarbons of the katydid and the odor of whatever volatile alarm pheromones it's releasing, in addition to a movement or vibrational cue produced by its struggles as it encountered the swarm. In contrast, the motionless dead katydid corpses presented the army ants only with the odors associated with the cuticle, the cuticular hydrocarbon odors of the dead katydid. And here's what we found in these two panels of this dot plot. Please note that each pair of black dots connected by a gray line are katydids paired by wet weight to control for the size and the magnitude, ultimately, of the stimulus being presented to the army ants. In panel A, we have recruitment rates. Panel B, we have mean running speeds of the Eceton burchellii. Again, these are proxies for the army ants' interest in these particular odor cues. And what we see is that Eceton burchellii detect and attack moving katydids, but ignore motionless katydid corpses, suggesting that it is not the odors of the katydid to which the burchellii is responding to, but the movement cues, the vibrational cues produced by a struggling, still living katydid, or non-ant arthropod, more generally. A few takeaways from the studies presented today show that two closely related arthropod predators exhibit variation in sensitivity to chemical and vibrational stimulus modalities relevant to foraging and prey capture. The similarities in chemical sensing by two different army ant species allow for a shared mechanism of ant prey detection and capture, but the mechanism has been honed to different target ant prey. Additionally, greater vibrational sensitivity allows Eceton burchellii to detect and capture large-bodied non-ant arthropods, a novel category of prey not taken by Eceton hematum or any other known Eceton species. This vibrational sensitivity may be a key adaptation allowing Eceton burchellii to be the only Eceton to consume a non-trivial amount of arthropods outside of the order Hymenoptera, thereby decreasing the likelihood of serious competition with its congeners. And to finish up, I have so many people to thank in my acknowledgements. Obviously, my PhD advisor and co-author, Scott Powell, for introducing me and encouraging me to work with these wonderful army ant critters. My STRI advisor, Bill Wieslow, my wonderful dissertation committee, Adam Smith and John Lill, the wonderful photographers, Alex Wild and Christian Ziegler, my funding sources, and of course, the residents of Barrow, Colorado Island. And with that, thank you for your attention. If I have any time left, I'd be happy to answer any questions.
Thank you, John. That was a great talk. We have time for one question. We have one question from Zach Phillips. Do prey ants of army ants use unattractive or odorless nest materials to conceal the odors of the nest materials that attract the army ants?
So while the studies I did didn't explicitly test that, we do know that from previous research that all nests or much of the nest material in ants generally does get pretty saturated, I guess is a good word, with the odors of the colony itself. They like a kind of a gestalt colony odor comprised of the cuticular hydrocarbons that ants will often use for things like nestmate recognition. So it would be really cool if we found a species of ant that was masking its cuticular hydrocarbons in some way to be avoided by army ant predation. But to my knowledge, no such ant has been found. I hope that answers your question.
Well, thank you so much, John. You were an excellent caboose to our session.
Thank you.
I want to thank everyone. I can honestly say that in all the conferences I've attended, I feel like this session had consistently just some of the best talks I've seen. So congratulations to all the presenters. Don't forget about those 3 virtual talks on the web. And